Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to International Conference, Faculty of Pharmacy, Universitas Muslim Indonesia. With a grand time, empowering natural product on drugs, discovery, and development. It is held to celebrate second in Indonesia, proudly present. The first Makassar International Conference in Pharmaceutical Science 2021. It's my pleasure to welcome you to this event. My name is Selby Andra Makfira, and I'll be your master of ceremony. Ladies and gentlemen, it's an honor to me to announce you that among us here, we have the Excellency Dean of Faculty of Pharmacy, Universitas Muslim Indonesia, Associate Professor Apoteker Rahmat Kosman, SSI MKes. The honorable speakers, Professor Abdul Munim, PhD, from University of Indonesia. Indonesia. Associate Professor Waranyo Pulcharon, PhD, from Chulalongkorn University, Thailand. Associate Professor Kyoko Nakagawa Goto, PhD, from Kanazawa University, Japan. Associate Professor Kenneth Terman Kongstad, PhD, from University of Copenhagen, Denmark. Associate Professor Dr. Shamima binti Abdul Rahman from University of Cyberjaya, Malaysia. The Honorable Heads of Faculty and Unit in Universitas Muslim Indonesia. The Honorable Vice Dean and Head of Study Programs in Faculty of Pharmacy, Universitas Muslim Indonesia. And for all distinguished guests and all the participants. Ladies and gentlemen, now we would like to invite the Honorable Dean of Faculty of Pharmacy to deliver welcome speech to Mr. Associate Professor Apoteker Rahmat Kosman, SSI MSI. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. The honorable speakers, the honorable lecturers and staff of the Faculty of Pharmacy, Universitas Muslim Indonesia, Makassar, the honorable oral presenters and all participants of the conference. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala asrafil anbiya'i wal mursalin Sayyidina Muhammad wa ala alihi wa sahbihi ajma'in. Ladies and gentlemen, on behalf of the Faculty of Pharmacy and organizing committee, we would like to welcome you all to the first Makassar International Conference on Pharmaceutical Science with the theme Empowering Natural Product and Drug Discovery and Development. We are holding this event to celebrate 20 years of the second decade on the establishment of the Faculty of Pharmacy, Universitas Muslim Indonesia Makassar, which is today, 26, 2021. Furthermore, I would like to express our appreciation to our expert presenters, Professor Dr. Abdul Munim from Indonesia University, Indonesia, Associate Professor Waranyo Polkaharun, PhD from Chulalongkorn University, Thailand, Associate Professor Kyoko Nakagawa Goto, PhD from Kanazawa University, Japan. Associate Professor Dr. Samima binti Abdul Rahman from Cyberjaya University, Malaysia, and Associate Professor Kenneth Terman Kongstad from Copenhagen University, Denmark, for your contribution in this conference. Also, to our co-host Faculty of Pharmacy, Cyberjaya University, Malaysia, for great collaboration. Finally, we hope you enjoy this conference. And please apologize for any inconvenience during conference. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Associate Professor Apotheker Rahmat Kosman, for a welcoming speech. Ladies and gentlemen, now we would move to the next event, International Webinar. For the first session, we would like to invite Apotheker Muammar Fawaz, PhD, to be the host. Mr. Apotheker Muammar Fawaz, PhD, the time is yours.
Bismillahirrahmanirrahim. At first, I'd like to say thank you to Master of Ceremony in our international conference. And also, I'd like to say I'm pleased to be here today. And good morning for everybody. I think you are so impressed to our international conference. And for the first session, I'd like to invite Professor Dr. Abdul Munim as first speaker. He is a lecturer in the Faculty of Pharmacy, University of Indonesia, and we can see his curriculum vitae. So, Professor Abdul Munim, PhD, is a lecturer in the University of Indonesia, and his focus research on the pharmacognosy. And so today, we hope we can get valuable information from him toward the pharmacognosy focus research. So, ladies and gentlemen, please welcome to the first presenter, Professor Dr. Abdul Munim. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Honorable Dean, Faculty of Pharmacy UMI Makassar, and Honorable Speaker of ICPS, also for committee. First of all, thank you for ICPS committee to invite me to share my experience in NADES study. So in this opportunity, I would like to present my research about utilization of natural deep protetic solvent in herbal product development. And the content of this presentation first is NADES, definition of NADES, and then utilization of NADES in pharmaceutical product development, and then NADES for plant metabolite extraction and prospect and challenges. So what is a natural deep protetic solvent, or NADES? If you see in internet or Googling about NADES, you will find this or some information about NADES. Some company in Europe, especially for cosmetics supplier raw material product, they sell some raw material in NADES solution. So you can see here about Naturex. They develop biomimetics botanical extract for cosmetic product with NADES technology, and then also NADES for microalgae biorefinery in cosmetic, and also the application of NADES in natural cosmetics. And also you will find some publication about NADES. This is one sample about NADES for improvement bioavailability pharmacokinetic of propanoid routine, distributed in proline glycine after administration in rats. So this is some information about NADES in internet. So now NADES get many attraction for researcher. So, and if you saw this slide, you will find the NADES publication increased rapidly in 2020. The publication is more than 150 publication, and if compared with another publication about extraction with non-NADES or in conventional method, the publication decrease gradually. So I think this is very interesting why the NADES publication increase rapidly. So what is NADES? This is graph about NADES. If you see in this slides, this is an illustration formation of natural deep protetic solvents, mixture of this is menthol and then lauric acid. Both compound is solid, but after mixing, they will be melt and to be liquid. So, and then as extraction solvent, we have to add water because the NADES consistency is very high, very viscous. So this is disadvantage of NADES. And if we see the temperature melting point of the component of NADES, we have two components: hydrogen bonding acceptor and hydrogen bonding donor. This is the melting point. One's about 80 and then another about 50. After mix, the melting point decrease until 20 degree. So the mechanism is like this. The hydrogen bondings about between HBA and HBD in this sample is 1,2-propanadiol with choline chlorides. And the hydrogen bonding can we evaluate in NMR to the NMR like this graph. So in the GC, we to evaluate the mechanism of NADES hydrogen bonding formation. So this the same phenomena. This is a formation of natural diprototic solvent through hydrogen bonding between choline chlorides. This is a hydrogen bonding acceptor, and then lactic acid as a hydrogen bonding donor. This is This is a hydrogen bonding in these yellow lines. And then this is some example of NADES component. One is a hydrogen bond donor. So you can see some or many organics, edible organics component like oxalic acid, malonic acid, and malic acid. This is carboxylic acid, and also the xylitol, and then glycerol. This is polyol, and then another component such as caffeic acid, gallic acid. This is a phenolic compound. So the component is edible and very safe to consume. And as a hydrogen acceptor, we can find some component such as nicotinic acid, betaine, choline chlorides. So kalau in our experience, choline chloride is better as a hydrogen donor acceptor. This is also is edible compound, and also you can see here some amino acid. Okay, and the next is slide is this is about utilization of NADES in pharmaceutical industry or in natural products development. One as plant metabolite extraction or secondary metabolites, and then from synthesis, medium synthesis, this can replace organic solvent, and then we can use this solution also to increase solubility or bioavailability of natural product because most of natural product has low solubility and low bioavailability, such as quercetin, and then curcumin, and then rutin. And also, we can increase the stability of pharmaceutical products, and some polyphenol has low stability and thermolabile. We can increase this system with NADES application. And then we can use this solvent also for micro extraction. This is extraction in small amount of sample. If we have only limited sample such as blood, so if we want to extract such as drug or their metabolite in limited sample of blood, we can use also natural dietetic solvent. And the last is a carrier in soluble compound, or this is we can use as also excipient or solvent in solution pharmaceutical solution product. This is some report about NADES to improve bioavailability of natural products. What is we can see here the application of NADES for extraction process and enhance in vivo bioavailability bioactive component from Cartamus tinctorius. This is the substance, maybe cartamin. So in this process, we can only use one solvent, natural deposit, as extraction solvent, and also they can act as enhancer for in vivo bioavailability. And the next is this is a review about application NADES to enhance solubility, dissolution rate, and oral bioavailability. So one solvent can enhancer solubility and dissolution. Also, same as or similar with Cartamus tinctorius for bioavailability in poorly water-soluble drug such as curcumin, andrografolid, and so on. And then we can see here NADES to improve bioavailability also from phenolic compound for blueberry. And also this is one process. The solvent, we can use this is as solvent in the end product. So we didn't remove the solvent from the product. And last is about this is bioavailability and the biological effect of and then hypothetic solvent microextraction for rhodamine, and then this is from researcher or from Turkey, the same group. They also use NADES for extraction of manganese. This is a good experiment, I think, because they use NADES for micro extraction or micro sampling of manganese, and then after they concentrate the extract, the determination of the metal, they use only spectrophotometry. They didn't use IAS. And same, this is also the same team. They use hypothetic solvent for micro extraction of the ethyl ethylcarbamate. This is maybe, I think, insecticide. So they successful to extract this insecticide from my environment product. I think I can see. So this slide, why? Okay, thank you. And then also this is NADES. We can use this solvent to increase stability of natural products isolation, because many of natural product has low stability and thermolabile. This is some natural products such as anthocyanin, tartamin, curcuminoid, phenolic. Many anthocyanin as reported, they extraction use NADES, and they successful to increase stability, because in our experience, anthocyanin only has stability not more than 1 month in a freezer. So the first is safflower. This is as bioactive compound is cartamine, and they use NADES, choline chloride, and glucose with optimum condition. They're using only heating for 30 minutes, 40 degree, and then Cataranthus or tapak dara in Indonesia, but they use the Cataranthus flower, and its target is anthocyanin, and the NADES is lactic acid, glucose 5:1, and the optimum condition they use ultrasound extraction for 30 minutes, and you can see also the other report, curcuminoid. They successful extracts or enhance the concentration of curcuminoid with citric acid and glucose, and the method is very, very simple, only stirring for 3 minutes, 30 minutes, 50 degree, and with 15% water addition. So this is a very, very interesting result. You can mix curcuminoid products in the end product as solution without you add emulgator and so on. This is a FTIR spectra of curcuminoid dissolved in NADES, and the interaction between NADES and curcumin. So this is, if you see here, you can see this is a IR spectrum, and the interaction or hydrogen bonding you can evaluate in this spectra. The red line is choline chlorides in middle spectrum, and then curcumin is blue line. This is a hydroxyl area. So after reaction between curcumin and choline chloride lactic acid, the hydroxyl group is disappear, the spectrum, and then the carbonyl group and benzene. This is a carbonyl group about 1,500 centimeter. Also, same after they form NADES, this spectrum also disappear, and this is a phenomena, and the interaction hydrogen bonding between NADES, choline chloride, and lactic acid with curcumin. Then this reaction product is soluble in water. So this is very interesting. This is carbonyl group. They disappear because they form hydrogen bonding, the bicarbonyl group with a hydroxyl group from choline chlorides. So very easy. You can use only ER spectrophotometer. And this is the same. This is a reaction between andrographolide from Andrographis paniculata. There's also dissoluble in water, but can solve in NADES. The NADES is lactic acid propanol. This is a propanol, and then propanol and lactic acid. This is the NADES can increase the solubility of andrographolide. This is the IR spectrum of same phenomena like curcumin. The hydrogen bonding between hydroxyl from androgapolid and then from lactic acid and carbonyl also, they can form bind with hydrogen bonding from hydroxyl group or carboxyl group. And then this is a publication as natural diptotic solvent as agent for improving solubility, stability, delivery of curcumin. So we can use only one solution, choline chloride and disolor, high potential curcumin dissolution. And then the system successfully applied an extraction medium from this nades for obtaining curcumin, and also as effective stabilizer to prevent curcumin degradation from sunlight. And the last, the solubility of curcumin is also increased, and bioavailability also increase after they make soluble in nades. So only one same component or one nades we can improve. First is extraction solvent, and then for increase their stabilization and also for the bioavailability, because we know the bioavailability of curcumin very low, only 25%. This is our experience about extraction of metabolite secondary with nades. The extraction mechanism, this is two mechanism for nades to extract metabolite from plant. One direct metabolite mechanism and indirect metabolism. The direct is interacting with target compound, usually through hydrogen bonding. This is we have see the mechanism in previous slide, and then the next is indirect mechanism through the imaging of the cell wall and then the target compound out from the plant matrix. So this is the mechanism about formation of hydrogen bonding between nades with target compound. This is choline chloride with maybe this is a carboxylic acid with target neochlorogenic acid. This is a formation of hydrogen bonding. And then in below, in this the same mechanism, but in this phenomena, also we can see about steric hindrance. So the steric hindrance is very important to get better extraction in metabolite secondary. So in the first step to get good solution of nades for extraction natural product, we have to screen the solubility of target in some natural diptotic solvent. This is a important step to screen the good or the best solvent for extraction of metabolite secondary. And this the phenomena about how nades can extract or can attract metabolite from cell. This is maybe similar with conventional method. The first, this is formation of nades, choline chloride with diptotic solvent. and then they penetrate through cell wall, and the target is this is phenolic compound or flavonoid, and then the NADES form hydrogen bonding, and the target is this is form a hydrogen bonding, and then after this formation and the concentration increase, they diffuse the product, and the product out from the cell. This is the mechanism extraction of flavonoid from cell using NADES via hydrogen bonding formation. And this is the same. This is bicalein as target. This is a flavonoid, and the base NADES is choline chloride with lactic acid or lefulinic acid. So maybe mistake. Maybe this is a lefulinic acid, and same or similar with curcumin and the andrografolate. We can evaluate their the formation about bicalein sulfation in NADES with spectrophotometer IR. This is a hydroxyl group zone, and also this will disappear after bicalein is make a solution in NADES. Also the carbonyl group, this is also disappear, and the ethereal group also disappear because the group or the part of this compound makes hydrogen bonding, and the spectrum disappear in or using only FTIR spectrum. And this is the second mechanism. The second mechanism with the cell wall destroying of liquids. You can see this is a same image of cinnamomum woodmanii box with NADES. The A, this is A. This is powder before treatment. The cell is very, very solid and very clear here. And after maceration, this is a conventional method, maceration using alcohol. Only a small amount of compound. This is extracted using alcohol, and the cell also still have good conformity. But after NADES extraction, this is C NADES extraction using choline chloride lactic acid, and then D is NADES choline chloride malic acid, and E choline chloride citric acid. So from this same image, we can see C is better solvent or better NADES because the liquids is very, very clear, and the matrix broken very bad. This is so the component or the matrix we can extract it very easy after NADES extraction. And the result from our experiment, NADES choline chloride and lactic acid is the best NADES for extraction tranexamic acid and coumarin from cinnamon buds, because the marker we use trans-cinnamaldehyde and coumarin in this experiment. And this is the same. This is the same image of peanut cell powder after and before treatment. A is powder before treatment, and B after maceration, and C after microwave extraction, and D NADES. We use ultrasound extraction with NADES, choline chloride, and oxalic acid, and E is ionic liquid. Ionic liquid and microwave extraction. Also, this is a green extraction. In this experiment, we use butyl methyl imidazole bromide, and also using the same ionic liquid, but we use treatment as a treatment or pre-treatment. We use urea, and the better or the best result is NADES if we compare with ionic liquid. From this image, also from analysis of resveratrol in peanut cell. And then this is some factor affecting result of extraction metabolite secondary with NADES. One is HBA, or hydrogen bonding acceptor, and hydrogen bonding donor. This is a type and molar ratio. This is one factor affecting the result. And then solid and liquid ratio, because NADES mostly is very viscous, and to good as solvent extraction, we have to add water. But if we add too much water, also is problem, because this is so it's not easy to evaporate the water from extract. And then same as a conventional water, time and temperature, and then application of addition energy such as ultrasonic, and then microwave extraction, and the last is mode of agitation. This is also same addition of energy to increase the result or optimum condition extraction. This is our research, some research about extraction of phenolic acid anthocyanin from flower of Ixora javanica, and this is the NADES, type of NADES, and then optimum condition, and the objective is to increase the metabolite. This is phenolic acid or phenol, and then anthocyanin, and the second is Morus alba as target oxyresveratrol, and the NADES is urea glycerin. This is very interesting, because in our experiment oxyresveratrol or Morus alba extraction, we will use this product as a cosmetic for whitening agent, and the urea and glycerin is excipient and good excipient for cosmetic as whitening agent. The objective same is to increase metabolite, and then green coffee as target caffeine, chlorogenic acid. This is the NADES, need to increase metabolite same. And the next is Leucania leucopalacids. The target is two component. One is sulfhydryl and then mimosin. But we know mimosin is the toxic product. So in this experiment, we want to increase sulfhydryl as a bioactive compound and to decrease the mimosin level. And we successful to reduce mimosin level and increase sulfhydryl component using choline sucrose with optimum condition. Use ultrasound extraction for 10 minutes, 75% water addition. And the last is cinnamon box as target cinnamaldehyde. You want to increase this component, and coumarin we want to decrease. Same as mimosin, this is also this very bioactive, but have some side effect. So same, we successful to decrease coumarin level and increase transenamyaldehyde using choline chloride and citric acid. This is the last experiment of our study about application NADES for extraction simultaneously using, this is a sapanwood and cinnamon. The target is a product for anti-diabetes, and the activity, we evaluate the in vitro activity using dipenyl peptidase 4 inhibitory activity. And the last is in vivo evaluation, but we have not carried out this experiment. This is introduction. Sapanwood and cinnamon have beneficial effect on diabetes mellitus, and then the mechanism via DPP4 inhibition. And also in this experiment, we want to extraction simultaneous of bioactive compound using NADES. This is the result. If we compare with conventional method, the NADES extraction very comparable with very best result. This is bioactive component, coumarin, cinnamaldehyde, and then as a marker from the sapanwood, the target is Brazilian. This is same. We compare NADES ultrasonic extraction compare with ultrasonic, but the solvent is water, and the result, the NADES is better than water. This is a SPLC chromatogram, and this is the inhibitor activity. The NADES very high activity and beta activity if we compare with reflux, and then we compare also with sitagliptin as positive control. Brazilian, this is as a standard for evaluation the DPP4 inhibitor activity. This is the IC50. We skip this. The next is bioactive technique from recovery. This is the problem because we have a problem to evaporate the solvent NADES for from the extract. So many method has reported to recover the bioactive from NADES. One is anti-solvent precipitation. We can use ethanol, methanol, ethyl acetate, and the next is counter-current chromatography or counter-current aspiration, and also we can use macrosporous resin absorption and solid phase extraction, back extraction, and LE ATPS. This is a method NADES using solid phase extraction. We can use cartridge with column cartridge here, the NADES, we can transfer the sample in here, and then elution, we can only use water, and then the target spirit, we can spirit from NADES. So NADES we can evaporate, and then we can reuse this solvent for the next experiment, and the target is very high purity. And the next is the application high countercurrent chromatography for simultaneous recovery or expiration of natural product from NADES. This is the flow charts of the expiration. And then this is a report from Su et al. About strategy for enhancement of phenolic acid extraction for mulberry leaf by natural diazotic solvent. And the recovery, they use macrosporous resin. This is a scheme of the expiration. And the last, we summarize some report about recovery bioactive compound from NADES using macrosporous resin. One is chlorogenic acid, caffeic acid from Lonicera japonica using NADES butanadiol choline chloride, and the recovery is about more than 80%. And then chlorogenic acid from Artemisia argyyi, the recovery is not good. And also we can see here about recovery of isoflavone, luretin, tectogenin, genistein, biocanin from Dalbergia odorata using NADES, and this is optimum condition for the recovery, and the results, the purity about 85%. This is the last of my slides about prospect and the challenging of NADES in the development of natural product. They have disadvantage also prospects. The NADES is have high density and viscosity if we compare with a conventional solvent. So addition water and increasing temperature can solve this problem. And the next NADES can extract not only target compound but also plant material such as protein, sugar residue, cellulose, etc. And low pressure of NADES makes difficulty of verification, so we can use another or other method to recover the bioactive compound. And some NADES were compatible with cosmetic application, pharmaceutical formulation, and for food manufacturing. And in cosmetics application, NADES can act also as excipient. This is good result from NADES application. Thank you. Terima kasih.
Thank you so much for your presentation, Professor Dr. Abdul Munim. And I think there are many questions from the attendees. At least there are 3 from the attendees, and I invite the first question from Mr. Ahmad Najib, because it is the live question, live interaction. So welcome to Mr. Ahmad Najib.
Okay, thank you very much for the moderator. Nice to see you again, Rob Munim.
Okay, nice to see you again.
Hopefully you and family. So it's a good presentation about NADES.
So maybe your mic's mute.
Okay, nice to meet you, sir. This is novel method to extract natural product, I think. So
It's not the sound is not clear
check,
for
check. Okay.
I can it's not it's
Okay.
still
Check.
not clear.
Test. Test.
Yeah.
Is clear? Test. Test.
Yeah.
Okay.
Unstable. Oh yeah. Okay. Okay.
Okay. Okay. I think this is quite presentation is very interesting because nowadays is a new method to extract a natural product. I've saying on internet there are new publication in 2020 until now, but I just want a big question: how to bring the lab scale to industrial scale to make, for example,
In commercial condition on the NADES products, we can find only in cosmetic raw material and especially from European country, the supplier, they sell product as liquid, so the handling also and distribution and the cost sure is higher than
yes.
solid or conventional extraction product, but they have the maybe advantage if we compare with conventional or solid, because the solution we can use as excipient, such as in our experiment, we successful in extraction of oxyresveratrol and resveratrol using NADES. The NADES is urea and glycerin. Urea and glycerin, this is also excipient in cosmetic products. So in the further process, we no need addition of urea. Urea is also can help to increase the activity as whitening, and then glycerol also they can act as a solvent, as pelembab apa tuh in English I lost. Yeah. So this is the advantage of NADES in cosmetic, but in another pharmaceutical product development, I have no information about the use NADES in another products, only in cosmetic. Yes, I sure this is the cost is higher than solid or in extract product using conventional, because they evaporates the solvent. Maybe this is challenging of the NADES products, but actually some supplier and from European country sell this product as liquid condition. Thank you, Pak Najib.
Thank you, sir. Thank you.
Professor Munim. And the next question from the chat, question and answer from the Faradiba, and she asks you how to know that the combination of this solvent is appropriate for our sample, for example, regarding the polarity. So what is your opinion?
Okay, thank you, Faradiba. So to get the best solvent, if we have many amount of standard of target compound, we can screen using such as if we have curcumin with pure condition, we can screen the solvent only. First, we mix some nades and then we add the curcumin with pure condition, and the best soluble curcumin is the juice, or this is the best nades for the extraction of the curcumin extraction. But if we have no much more standard in our experience, I just we search the literature and we compare the structure of our target with another report, and we can use some NADE solution from literature review, because the parameter not only polarity. If we see the mechanism, this is hydrogen bonding. So if hydrogen bondings formation, theoretically the extraction or the target for also solar polar target. But some report curcumin, andugrapolid, and also steroid reported that can extracted using NADES. So if you want to fast experiment, you can search the literature review and you show only the similarity of target compound structure. Maybe it's clear.
How to choose the combination of solvents, and we can see the previous paper are based on the structure of the target compound. So the next question from the same attendees, how to make a combination. I think you already answered this question based on the previous paper, a review paper, or based on the structure of target molecule. So I'm going to the next question because we still have 10 minutes. So the next question from Andi Rismayanti, she asks you about the application of nano extraction, how to apply the nano extraction in the nanoparticle medical technology, maybe related between the nano extraction and nanoparticle medical technology.
Okay, the nano extraction method for nanoparticle product, maybe this is because I have no experience of this experiment, but in our opinion, we can extract from we see the previous literature, some micro extraction, nano extraction also have reported, but in the problem, the extraction we have to optimize because we need the addition of such as anti-solvent. Some report, if you see in literature, the first step is the addition of anti-solvent. So anti-solvent, this is working to break the hydrogen bonding, such that we can use ethyl acetate, and then benzene, also methanol, ethanol. After that, we use or we add the NADES, suitable NADES. So we have two steps. First is addition of antisolvent, and then the last is optimum condition of NADES extraction.
So based on your experience or your opinion, we can also use the combination of NADES to extract the nanoparticle product from the, the nanoparticle product.
Yeah.
Yeah, okay. So the next question from Rianti Anissa, we still have 5 minutes. About the product from the NADES as a solvent, affecting the compound could be mono or poly compound. So I think you already show us at least sulfhydryl and mimosin. The sulfhydryl, the NADES could increase the extraction of sulfhydryl and reduce the mimosin. So how you explain that?
Okay. For extraction of NADES from natural product target in our equation, we can selective extraction. So this is need more experiment, because in the first, if we want to extract selective, we have problem. The natural products in plant, they have such as they have some group of components such as amidine, alloamidine, and then the same or the similar of group of amidine. So this is the compound. If we see the structure, the structure only a little bit differentiation. So it is very difficult to select if only one type of or one compound. But if we want to get whole anthraquinone and another group not extracted, is very easy. So the problem, if we want to extract such as curcumin 1, curcumin 2, curcumin 3, yeah, it's easy. But if we want to only curcumin 1, is in our experience, I'm not successful to get only one component. So maybe we need some or many solvent to screen the optimum or suitable component of NADES. This is the problem in extraction of natural product, because many natural product is in one of a group such as alkaloid and IBCD and anthraquinone, also curcumin, and so on. This is in our experience the problem if we want only one target. But we can solve the problem if we want to recovery. We use such as chromatography. In our lab, we know how to extract and then separate the component of zingiber, such as Sagol A, Sagol 6, Sagol 8 and 10, also gingibrol 6, gingibrol 8, and 10 using NADES. But after that, we use CPC, centrifugal partition chromatography. So we have to use more than one method. This is our experience.
Yes, I think this discussion is my cup of tea. So I think this time is not enough to discuss many things. So we can make a correspondence with Professor Abdul Muin for the next time. So I just want to make a summary for this topic. At least there are 4 function or of the natural deep eutectic solvent NADES. The first one is to improve the extraction of target compound, and at the same time to reduce the untargeted compound, and also to improve the stability of targeted compound and improve the bioavailability of targeted compound. I think that's a point I can summary. So thank you so much for your presentation, Professor Abdul Munim, and see you later.
Moderator.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
So the next session, I invite the next presenter, Associate Professor. Maybe she already joined in our webinar. Associate Professor Kyoko Nakagawa Goto, PhD, from Kanazawa University. She is a researcher and lecturer in the Laboratory of Pharmacognosy, Molecular Pharmacognosy, Kanazawa University, Japan.
I thank you for your introduction. I will share the slide.
So welcome to Dr. Goto, or we can say that
So can
Goto Sensei
from
the Kanazawa University, Japan. Time is yours.
Okay. Thank you for introduction. You can hear me or you can see?
Yes, I can see your screen.
Okay. So thank you for giving me an opportunity to talk about our research. Today, I would like to talk about our recent achievement entitled Drug Discovery and Development Based on Natural Products. Our ultimate aim is to find drug candidate with first-in-class drugs. Let's think about the current problem for drug discovery. This graph shows the number of approved drugs by FDA each 5-year group. The red circle shows the number of total approved drugs, and the black square shows small molecule drugs. Before 1990, the phenotype-based drug discovery, we call it PDD, was conducted using mainly natural products. After 1990, the scientific technology was remarkably developed, which induced the combination of combinatorial chemistry and high-throughput screening, including virtual one. Despite that, the number of new drugs has not increased, as you can see. One of considerable reason is the lack of chemical diversity in our chemical library. And most drugs target the limited protein as shown in the next slide. The left figure shows that only small fraction here of total protein were targeted for the current drug. This indicates that we have a lot of undiscovered drug target even now. Since the protein has a diverse 3D structures, we need to expand the chemical space to find a new drug target. The figure on the right side shows that the predicted total chemical space and the chemical space occupied by natural product and synthetic drugs. As you know well, natural product has a wide range of chemical space compared with the synthetic one. So we think that the use of natural product combination with chemical modification provides the expansion of this chemical space. Altogether, the forward pharmacology approach, which combines phenotype drug discovery with chemical modification of natural products, is well suited for finding first-in-class drugs. Here is a summary of our outline for our PDD approach. Bioactive natural products, I will say BNP from now, can be found from tropical plants using the knowledge and techniques of phytochemistry. The establishment of total synthesis following chemical modification will be carried out using organic chemistry. Synthesized compound will be evaluated for biological activity. We are currently focused on anti-tumor and antiviral activity. The next study of structure-activity relationship, SAR, is very important to know variable pharmacophore to develop more better drug candidate. This requires medicinal chemistry. Next, the crucial study for mechanism of action, MOA, and target identification can be performed using various techniques such as flow cytometer, cell immunostaining, cell-free assay, computer modeling, as well as affinity verification. Using these methods, we try to find a candidate for a first-in-class drugs with novel targets. Currently, 3 main projects are now ongoing in our group. First, isolation and identification of BNPs from natural products should have high affinity for the enzyme or protein, which are related to their bioactivity for other species. In most cases, the basic skeleton belongs to privileged structure, which are defined as the molecular frameworks for useful drug design. Using natural skeleton, we try to create a novel bioactive compound by insertion of non-natural unit to the skeleton. I would like to talk about the first project mainly, and briefly second and third project today. As I mentioned before, we focused on the plant in rainforest area. Tropical rainforest is located around the equator. These areas are generally hot and wet all seasons, which have made a high level of biodiversity as described in this slide. The most important fact is 25% of drugs have been discovered from organisms living in the rainforest. Thus, tropical rainforests have been called the world's largest pharmacy. Unfortunately, 50,000 species of plants, animals, and insects disappear annually. This area is currently in danger of extinction. Although only few % of plants have been investigated phytochemically so far. Thus, we need to perform the phytochemical study in haste. For our first project, we used 2 resources to get the rainforest plant extract. One is National Cancer Institute, NCI, in the US. NCI stored 80,000 species of plant extract collected from around the world in 1990. Among them, about 3,000 species showed anti-tumor activity after NCI 60 panel screening. Think about how many plant species are from rainforest area among them. Surprisingly, almost 70% of species were collected from rainforest area. In fact, many anti-cancer drugs, which are currently clinically used, were found from these plant extracts. However, they have still many unanalyzed extracts. We have collaborated to two key persons, Dr. Newman and O'Keefe, to find additional potent bioactive compound from these plant extracts. Indonesian plants are also important for our project. Indonesia is known as a mega diverse country because it's located near Wallace Line. We have collaborated with Dr. Alam and Rahim in Hasanuddin University. Level 1 and Level 2 shows our research using NCI plant and Indonesian plant extract, respectively. Some of them are currently in progress. The figures on the left side showed some of isolated compound from NCI and Indonesian plant, respectively. Today, I would like to talk about the 3 plant extracts from NCI, entry number 1, 2, and 4, as well as Indonesian plant extract number 9 as our isolation project. Let me talk about the 50% methanol in methylene chloride extract of Rhaetia colibrosa. The extract showed potent anti-proliferative activity by NCI 60 panel screening. The activity was further confirmed in-house assay system using 5 human tumor cell lines as shown here. The results also showed significant activity as shown in this table. After purification using various techniques of column chromatography, 27 of clerodine diterpenes, including 20 new ones, were isolated. Their structures commonly have tetrahydrofuran at C4, C5, and acetate at C18 and C19, as well as a branched C7 unit at C9. The differences are only the stereochemistry at C2 and the function group at C2 and C6 position. Most cases, the absolute configuration of this type of compound were not determined in the previous studies. In this work, we determined the absolute configuration of all isolated compounds through chemical method or ECG experiment, comparison of spectroscopic data, and X-ray techniques. All isolated compounds can be classified into 5 groups from their difference of stereochemistry at C2 and function group at C2 and C6. The compound in group A have 2 hydroxy and 6 ester group, as well as R configuration at C2. The compound in the group B have the same substituent pattern at C2 and C6 as those of group A. However, C2 is S configuration. Activity of these groups showed interesting SAR as shown in the next. Here is the activity for the selected compound. The activity is shown in IC50 value. Oh, this is nanomolar, I'm sorry. this is mistake. Unit is nanomolar. Among them, compound 6 in group C show the weakest activity. And the compound in group A and B, namely 2-hydroxy 6-ester analogs, displayed mild activity. Compared with them, the most 2-ester 6-hydroxy analogs in group D and E showed remarkable activity with IC50 value of under 1 micromolar. In this group, 6-hydroxy is preferred to 6-hydroxy is preferred to 6-methoxy group. When the comparison of compound 7 versus compound 9 and compound 12 versus compound 15. Although compound 11 and 14 shows similar activity. This table clearly shows that the stereochemistry at C2 is not important for the activity. Here is the second plant, cryptocarrier ligator, which is also provided by NCI. The result of in-house anti-proliferative activity is shown in the top here. From this plant, we have isolated the very unique meroterpenoid with spiral non-linear skeleton named cryptolive lactons A to L. The structure is divided into 2 portions, spiral moiety and lacton moiety. The difference of spiral moiety is the stereochemistry at C11, 12, 1', 4'. The table in the center shows this one, their structure. For example, cryptolive lacton A, we call it CLA, consists spiral part X with lacton part R1. Lacton moiety R1 and R2 are slowly converted to the related R3 and R4 by Michael cyclization, respectively. Along with these spiral compounds, a linear lacton CLM was also newly isolated. The structure was also confirmed by the asymmetric total synthesis starting from 1,3-propanediol through Keck and Brown asymmetric allylation to construct the C6, C8 alcohol following Grubbs olefin metastasis to form lacton and Grignard reaction with sterile magnesium bromide. Currently, the total synthesis of CLA to L are in progress to confirm their stereochemistry and to evaluate their biological activity. The proposed biosynthetic pathway is described in this slide. After formation poly-beta ketone chain through well-known pathway, the intermediate B could be constructed. Dehydration of 4-hydroxy and dehydroxylation of 12-hydroxy formed CLM. On the other hand, dehydration of 12-hydroxy group could obtain alpha-beta unsaturated ketone, which could be reacted with beta-pherandrine, which was also isolated from this genus. The 2+2 cyclization could form spiral 3,5 nonlinear structure to produce CLA to C and L. Following Michael addition could give bicyclic pylon CL D to F. A saturation of ClB would give ClK. Dehydration of the related Cls would give Cl2G2L. Next, in-house cytotoxic assay of the plant extract of Acronychia triflorata showed almost 100% growth inhibition of tumor cell line at 20 microgram per milliliter of concentration. Despite only 5 grams of extract, we have succeeded to isolate 20 prenulated acetophenols, including 7 new ones named acronycracin I2O, one known monomer which was revised the structure, together with 4 known monomers. Along with monomers, 3 new dimers and 5 known dimers were also isolated. Especially compound 13 is very unique dimer. Only 4 related dimers without prenyl group were found previously. Because of very limited amount of compound, we couldn't evaluate for any bioactivity for these compounds. Thus, we started the total synthesis of them, which is a part of our project number 2. To confirm the structure and evaluate the biological activity, the isolated monomers and their analogs were synthesized starting from trihydroxyacetophenone THA, as shown here. In the interest of time, I will skip the detail, but we have succeeded the synthesis of the desired acetophenone monomers. All physical data of synthesized compounds were consistent with those of the natural product isolated from the plant. None of acetophenone monomers shows potent anti-proliferative activity. However, we have found that the compound with prenyl-like function group showed significant tumor prevention effects. Acetophenone dimer and some analogs were also synthesized as shown here. The acetophenone dimers were evaluated for anti-tumor proliferative activity as shown here. Many of dimers showed potent activity, although the monomers did not. Most active compounds have a prenyl-like function group as shown here. This result indicated that prenyl-like function group might be suitable for the activity. The last one for project number 1 is an Indonesian plant, Melonica umbellata, which was collaborated in Sulawesi Island. Which was collected in Sulawesi Island at Indonesia by collaboration project with Hasanuddin University. From this plant, 5,3,4-methylene fused with phenyl 14 oxabicyclose 321 octane were obtained as novel compound named pariasanin A to E along with some quinolones. The anti-proliferative activity of isolated compounds are in this table. The result indicates that methoxyquinolone skeleton, this type, is favored for the activity rather than methylene dioxykitolin skeleton. As a result, valsartan A and its methyl analog showed selective activity against lung cancer cell line A549. Very recently, we have achieved the total synthesis of varicellin A, and currently chemical modifications are underway. This slide showed our second project. Let me explain very briefly. The first one is an unusual flavonoid with non-aromatic ring A, dysmos-demotin B, isolated from dysmos-demosas. After achievement of total synthesis, over 50 of analogs were synthesized and evaluated for anti-proliferative activity. As a result, we successfully improved the original activity, which shows selective cytotoxicity against only MDR tumor cell line by changing the function group on the ring A. In addition, the replacement of phenyl ring B to benzothiophene displayed dramatical change of biological profile, showing significant cytotoxicity against all tested tumor cell lines. We also tried to identify the target protein with various techniques as shown here. The second one is terpenoid, isolated from Plectranthus parvuloflorus. After achievement of total synthesis, we have found two related analogs showed selective anti-tumor, anti-proliferative activity against triple-negative breast cancer, TNBC. We performed affinity verification and caught up two target proteins. Currently, the confirmation study is ongoing. Here is an example for our third project, which is focused on the natural product skeleton, not BNP itself. Using chromon, which is one of the natural product skeleton, we successfully create the new anti-proliferative compound by the replacement of phenyl ring B to benzothiophene. The compound induces cell cycle arrest at G2M phase and inhibited tubulin polymerization. Interestingly, small modification here and here induce the different biological profile, showing the inhibition of topoisomerase 1. These results were reported in this journal very recently. Our recent achievement for project number 2 and 3 are described here. Chalcone, diterpene, sesquiterpene, flavonoid, lignan, alkaloid, and dialheptanoid. If you are interested in our research, please use this QR code. You can see our achievement more detail. At the end, I would like to acknowledge these persons. Our works were not accomplished without their help. In addition, these works were supported by 4 grants as shown here. I also say thank you to all of my lab members, as shown here. Thank you for your attention. I'm done. Do you have any question?
Miss Faradiba?
Thank you, moderator. Ohayo gozaimasu, sensei.
Ohayo.
Genki desu ka?
Genki desu.
Alhamdulillah. Sensei, I want to ask some question. First, I have seen many of sensei research about anti-tumor and antiviral. And the question is, in sensei research, have find many structure of this BADP or bioactivity molecule compound. I want to ask if whether there is a one or same structure of this structure you found that this structure is specific for anti-tumor or antiviral from all of the sensei research. If there any specific structure that sensei found in this molecular structure that we can develop for anti-tumor for the next anti-tumor. This is the first one. And the second one, in this design and synthesis of this molecule, we found that sensei added with benzopyrene to modification the structure. In sensei opinion, this is benzopyrene could be possibility development for anti-tumor drugs for the future. That's all, sensei. Thank you very much, moderator.
Okay.
Thank you.
I don't understand well, but first question, you want to say how to predict the target. If the target is different, the structure also different. So it's hard to predict, but we can estimate when you search the literature. This type of the structure may have the anti-tumor, or this type of structure has the antiviral or something like that. But it is very hard. For example, in our research also showed, if they have the same structure, it depends on the function group. It's different. The activity will depend on the function group. So it is very hard. If you have identified the protein, target protein, you can use computer modeling or something like that to estimate the structure. But usually it's very hard. And the second one is benzothiazine. Second one. What's the question? Second one. Say it again, please.
Ms. Faradiba, could you please repeat your question for the second one?
Excuse me, Sensei. I see in Sensei research about synthesis and design of the drug. Sensei added with the benzopyrene, the modification of the structure. Is there? Yes. I see that is benzopyrene is potential for developing the cancer from the plant, Sensei. After we isolate it and then we make a synthesis and then design. Oh, that's all, Sensei. Thank you.
Okay, in our compound, the important thing is the tempi electron system. Benzopyrene is not tempi. So benzofuran, I try to make a benzofuran or naphthalene. However, the most important compound is benzothiazine. It depends on aromaticity of the compound.
Okay. So, is it clear?
Okay, Sensei.
Okay,
thank you, Sensei.
Okay, I think it's clear, the explanation from Goto Sensei. So, I invite the second questioner from the Rizky Wahyuni Aris. Miss Rizky, can you hear me? Hello? Oh, okay. So, okay. Miss Rizky, time is yours, please. Okay, I think Ms. Rizky is not in our Zoom webinar, so next I invite all participants to give a question. We have 10 minutes left for this session. Okay. So about your presentation, you isolate the compound from the prospective natural compound for example, from Sulawesi, Indonesia, and also you synthesize because of the lack or the problem to isolate from the natural product. So what do you think about the perspective of this topic or this focused research for the future?
Future. If we can find the potent drug seed, we want to, if possible, we want to ask some pharmaceutical agent to develop the clinical drug. If possible.
Okay.
But it's very hard.
I think it's a good idea, Sensei. So I think today there is no question anymore from the participants. So I'd like to say thank you so much for your time and your presentation. Also, I'd like to say thank you to all participants for their active participation. And I hope today the presentation or the topic will give us the benefit for our life or for our future. So thank you so much for your time and presentation, Goto Sensei. Nice to meet you and see you later.
Thank you very much. See you later. Bye.
Bye. So the next session, the second session will be held by Miss Nurmaya Effendi, PhD, as the moderator, and they will be a moderator for two presenters. Okay, thank you.
Tes. Kenapa ini? Oh, oke. Tidak ada host kok.
Langsung mungkin.
Oke. Can I start? The voice is okay? Masuk atau enggak? Saya enggak tahu.
Warnanya. Karena dia keluar.
The voice is okay? The voice is okay? Okay. Dr. Sahima. Dr. Waranyo, do you hear me?
Hello, yes, I can hear
Welcome.
you. Hello, Asa.
Dr. Sahima, okay. Okay, thank you. Now we are in the the third section. Okay, let's start. Before that, I want to say on behalf of University Muslim Indonesia, two speakers who is share with us about their research. The first one is Associate Professor Waranyo Pulcharon. I'm sorry if I made when I'm spelling your name. Is that's right?
Yes, that's right.
So sorry. Okay, can I call you Dr. Waranyo? It's okay?
Yes.
Oh, okay, thank
Yes.
you. How are you?
I'm good, thanks.
Thank you. Being willing to be speaker today. Thank you. Maybe you are the first speaker in this section. And before that, before Dr. Waranyo start with her presentation, may I introduce her. Associate Professor Waranyo Pulcharon. She is one of lecturer at Faculty of Pharmacy. Sorry. Give me a minute. Okay. Dr. Waranyo is one of lecturer at Faculty of Pharmaceutical Science, Chulalongkorn University since 2011. She has from international supporting by this grant for his research, her research, And then, I think it is enough for introduction for Dr. Waranyo. So I think, are you ready, Dr. Waranyo, for presentation? Okay,
Yes.
the time is yours.
Can I start sharing my screen? Okay. Okay. Can you hear? Can you see my slide? Okay. Good morning, everyone. First of all, I would like to thank the organizer for inviting me to join the conference today and to share my research. Right now, I am, as the moderator introducing me, I am working in Faculty of Pharmaceutical Science, Chulalongkorn University in Bangkok. And my research area is on producing recombinant proteins using plant as the platform. Right now, we have the startup company, which is the spin-off company from Chulalongkorn University, and the company is Bayer Phytopharm. This is like the new policy for university to push the faculty member to use their research experience to have commercialize the product. And we started a company for a few years, and today I would like to share you about the molecular farming, and this is the platform that can produce new biopharmaceuticals. Then to use plants to produce biopharmaceuticals, we would like to have replace the using of fermenter. If you familiar with recombinant protein production using in the drug industry, most company they use mammalian cells, bacterial cells, yeast, or even insect cells. When you use the cells to produce recombinant proteins, you will familiar with the left figure. You can see the big fermenters, right? But with this technology, you have to use the fermenter, and even the media for the cells is very expensive. For Thailand, if you want to have the industry use this kind of technology, the fermenter you have to order from abroad. We cannot produce in our country. Even the media for the cells, if you are familiar with the research that use the media, you know how expensive it is, right? And we have to import as well. Then that's why we think that if we can change the way to use this kind of fermenter, to use the right figure, to use whole plant as the factory, it would be quick for us. If we want to produce more protein, we can just grow more plant. And I think for Thailand, even I think for Indonesia or many countries in Southeast Asia, I think we have the capacity for growing plants, right? Even the salad, people sell the plant, they can do hydroponic plants, they can grow more plants. Then we think that using the plant as the platform to produce biopharmaceuticals, this is kind of the cutting edge solution that can produce new biopharmaceuticals that is quick, simple, and scalable. If we want to produce more protein, we just grow more plant for that, right? And this is going to be the faster and lower cost of research and development for the investment. The recombinant proteins from plants, we can apply to several applications. If we all work in the lab, we have to use the reagent that is recombinant proteins. For example, people working with the cells, you use mammalian cells for testing all the compounds. You have to use some kind of growth factors. These kind of growth factors we can use the plant as to produce as well. We can produce the growth factors for cosmetic materials. Right now there are several there's new trend of the cosmetic use instead of using like herbal products. Several companies changed to use growth factors that produce from plant. Okay, and we can apply the proteins to use as the diagnostic kit. You know, like the to test diagnostic kit to test for different disease. And the final goal for us is to use these plant-produced recombinant proteins as the biologic drugs and vaccines. We start the Bayer Phytopharm for 3 years. You know, like the company start for 2018. Before that, we did like all research in the lab. You know, and we just published several papers on plant-produced vaccines or plant-produced therapeutics proteins. Okay, but after we start the company, first thing we have to try to scale up, like how to grow more plant and how to produce more proteins. That's the first aim that we did, and there's two of the co-founders for Bayer Phytopharm. Dr. Suthila is CEO of the company, and myself, I am the CTO of the company. Two of us are the faculty members in Faculty of Pharmaceutical Science, Chulalongkorn University. We have several products in the pipelines, like we have growth factor, human growth factors. We start developing the diagnostic kit, the test kit for hand, foot, mouth disease. You know, in Thailand, every year we have the outbreak of hand, foot, mouth disease for the kids, and the school closed. You know, and that affect the parents. You know, then we think that if we can detect the kid that affect with hand, foot, mouth quick, then we can have quarantine the kids quicker, which will prevent the outbreak of the disease. That's one thing. We have the drug for animal and also for human in the pipeline, you know, but after we start the company in 2018, and then, you know, like around 1 year, at the end of 2019, COVID came, you know, like, and that affects everyone. I think like no one don't know COVID-19, right? When we have the COVID-19 to our countries, then, you know, like we have we cannot verify our hand, foot, mouth disease, you know, because everyone's like stay social distancing, wearing masks, you know, and no case for hand, foot, mouth disease. Then we just stop that, you know, we just stop all the project, and we try to think what can we do with what we have to do something for COVID-19. Okay, then we just stop doing all of our other proteins, and we produce all of the proteins involved related to COVID-19. For example, we start producing several monoclonal antibodies against COVID-19, and, you know, like we try to test whether it can neutralize the virus, you know, and we also produce several types of the protein that is the part of the virus itself, and we plan to test it as the vaccine. Okay, with our platform, we can show that we can produce new recombinant proteins very quick. You know, like after we get the genes that we synthesize, we found that we took only 10 days, and we can produce the proteins. Okay, then many of you might have the question how we produce the recombinant protein rapidly. Okay, you might familiar with the way to produce recombinant proteins using transgenic plant, right? For transgenic plant, it takes long time, like at least 6 months to produce new protein, right? However, the technology that we use in our lab and in Bayer Phytopharm, we use the techniques called transient plant-based expression. And for these techniques, what we do is we use the plant. This is tobacco plant. The scientific name is Nicotiana bentamiana. Okay, and the plant that we grow, this is wild type plant. we did not genetically engineer the plant, like this plant is like wild type plant. And after we get the size of the plant that we want, what we do is we just transfer the gene that we are interested in into the plant by using agrobacterium. Agrobacterium is bacteria in nature that can transfer the gene into the plant. Okay, what we do is, like, let's say, for example, if we want to produce the spike of the virus, okay, we just insert the gene, the spike gene of the virus into agrobacterium, okay, and we infiltrate agrobacterium into the plant, okay. After agrobacterium get into the leaf, it can transfer the gene into the plant cells. And after that, we just grow the plant for like more 3 to 7 days, only short period. In this 3 to 7 days, the plant can use their transcription and translation. Then they can produce the spike protein that we insert the gene into it. After that, we just harvest the plant, like cut the whole plant, okay, and extract the protein. Then in the protein extraction, we get all of the plant proteins with the protein that we are interested in, which is the protein that we insert the gene into the plant, right? Then we have to use this protein extract to go through protein purification step. In this protein purification step, we remove all of the plant proteins out, and we get only the protein that we are interested in. In this case, is the spike protein, right? And then after protein purification, we can get only the protein that we are interested in. Then you can see that the whole process, it takes like less than 10 days. That's why we can get the new recombinant proteins in 10 days. And in this case, if we want to have more proteins, we just have to use more plants, right? And then we can get more proteins. For COVID-19 vaccines, the protein that we produce, we call it subunit vaccine or the protein vaccine, right? Then the left figure show the diagram of the SARS-CoV-2, and you can see the red spike on the envelope of the virus. This spike protein is the target for the vaccine candidate. We know from the SARS-CoV-1 that this protein is has potential to use as the effective vaccine. Then we produce this spike protein. We have several types of the protein. We produce whole spike. We produce only S1, which is subunit vaccine, and we also produce only small part, which is RBD or receptor binding domain. Okay, because from previous study, they show that only the RBD can induce neutralizing antibody. Okay, then after we synthesize the gene, we clone this gene into plant expression vector, and then we transform this into agrobacterium. After agrobacterium infiltrate into Nicotiana pentamina plant, it transfer the vector into the leaf, okay, and this the agrobacterium can transfer the plasmid into the plant cells. After that, we grow the plant for 4 more days, and during these 4 more days, plants can produce more protein, okay, and after that, we can extract and purify the protein. On the right figure, you can see that we can get the purified protein. Okay, like the figure B show the purified protein in SDS page, and you can see that we can see the reducing gel show the bands almost like 75 kilodaltons, and non-reducing gel show the dimer, the size around 150 kilodaltons, and the figure C show Western blot, like we confirm the expression of our protein with the monoclonal antibody against receptor binding domain, and this confirms that we can produce the RBD protein of the SARS-CoV-2 virus in plants. Okay, after we get the purified protein, we did test our protein in mice, and this is the immunization schedule. We immunize IM of our vaccine to the mice for 2 doses with 3 weeks interval. Okay, we get the blood like 2 weeks after immunization. And if you see the top left figure, this is the total IgG. They show that our protein RBD-FC itself can induce immune response. However, when we immunize the mice for 2 doses, we get higher antibody titer. We also analyze for IgG1 and IgG2A, and also the bottom right show the neutralizing antibody. I think like the neutralizing antibody is the antibody that can inhibit virus infection. And from this figure, you can see that the protein itself, after 2 doses, it can induce neutralizing antibody. However, our protein with alum as an adjuvant can induce high neutralizing antibody titer after 2 doses. After we get this result, we continue to study our vaccines in the monkeys, okay? Because monkey is non-human primate. It is the species that's closer to human. We use the same schedule for the monkeys, and you can see that in this one, we increase the dose from 10 microgram in mice to 25 and 50 microgram, and we did immunize IM in the monkeys. Okay, 2 doses, the same with 3 weeks interval. What we see is that for the antibody titer, both 25 and 50 microgram of the protein with alum can induce high antibody titer. And we also get high neutralizing antibody titers. Okay, and after we saw this data, we did another immunization, another experiment, and decrease the dose to 5 and 10 microgram of the protein. And what we found is that our vaccine 5, 10, 25, and 50 microgram, we get the same immune response. And it seems like the immune response is dose independent. Okay, then right now we get quite exciting data from both mice and monkeys. Okay, also as we all know that right now the virus mutate and we get several variants of concern, right? Then we did test our vaccines, whether it can induce the antibody against other variants of concerns. And what we found is that our vaccine can induce a good immune response against Wuhan and Alpha strain. With Delta, we still get high antibody titer, okay? But the antibody titer against Beta and Gamma is not that good, you know, like you can see the level is dramatically lower. Then after we did like 2 immunization, we have the monkeys, like we still keep the monkeys, and after 4 months after the second dose, we did try to boost again with the same vaccine. What we saw is that after 3 doses, the vaccine can induce immune response against all of the variants of concern. You can see that we can get the higher immune response against Beta and Gamma, right? Then this is like quite exciting data that we get right now. Also, in the same time, our research and development team still not stopped, you know, like we still produce some other proteins for different variants, you know, like this paper show that we produce the RBD proteins for alpha and beta. And right now we show that we can produce this protein. Okay, right now we have the RBD for almost 10 variants. Okay, and we are still keep testing whether this vaccine is effective for this variant of concerns. Mm-hmm. And as from last year, when we know that we get quite good data for our plant vaccine efficacy, then we start planning for doing clinical trial in human, clinical trial phase 1 to test the safety of our plant-produced proteins. And then from last year, we start building the facility, you know, the GMP facility for producing plant-produced vaccine that can test in human. Okay, then we just produce the facility that we can grow plants, we can infiltrate and extract. However, with COVID-19, time is one limiting factor, and we don't want to like build the whole process in our facility. Then we try to find a partner in Thailand. We have Kinjen Biotech. They have the facility for purifying protein already, which is a GMP facility. And then what we plan is that we can grow the plant, infiltrate and extract, right? And then we can send the protein extract with good transportation practice to Kinjen. They can purify the protein, and then they can send, after we get the purified protein or the drug substance, we can transfer this drug substance to Thai Red Cross, which they have the facility for formulating and filling. Then we have like 3 facilities working together on producing the vaccine for clinical trial phase 1. Okay, and right now we finished our facility for producing plant-produced vaccine, and this is the first plant-based GMP manufacturer in Thailand and even in Southeast Asia. I just want to show you let me show you the facility, okay? But the video is in Thai, but I will explain. I just want to show you to see how the okay. Can you see? Okay, then this is the facility that we built in Chulalongkorn University. We have the clean room that we grow the plants here. Okay. This facility is not only for COVID-19, but we can use it to produce other vaccine as well. Okay. Then we grow the plant here on the 11th floor in the building in Chulalongkorn University. We start seeding and growing the plant in this facility. We have around 200 square meters for the plant. Okay. Then we have to do the seeding, grow the tobacco plant in the room that we control environment, control the temperatures. Okay. After we infiltrate, we extract the protein here. We get the crude extract, the green juice, and we send these to Kinjen Biotech for purification. Okay. And send the protein to formulate in Thai Red Cross. With this facility, we can produce the vaccines around 1 to 5 million doses per month.
และที่นี่คืออีกหนึ่งความหวังใหม่ของการขับเคลื่อนเศรษฐกิจไทยให้กลับมาหยัดยืนได้อีกครั้งขอขอบคุณทุกแรงใจจากคนไทยที่ได้ร่วมเป็นส่วนหนึ่งของทีมไทยแลนด์อีกไม่นานเกินรอฝันของคนไทยกำลังใกล้จะเป็นจริงวัคซีนเพื่อคนไทยวัคซีนจุฬาไปยา
Okay, then you can have some idea how the facility look like. Okay, and then not only the COVID-19 vaccine, you know, like as I told you, we can produce other type of the drug or the vaccine. You know, one thing that we are also developing is the monoclonal antibody for cancer immunotherapy. Now we produce the drug named nivolumab, which is the anti-human PD-1, and we did test in the mice. You know, like right now we got the data. We saw that you know, like when the mice have the tumors, you know, like you can see the red graph is the Opdivo, which is the commercial drug, and the blue one is the plant-produced nivolumab. And you can see that the plant-produced monoclonal antibody can decrease the tumor size in the mice. Then this is like another product that we are in the process of developing, and we hope that next year we can push this product to clinical trial phase 1. And you know, like for the COVID-19 vaccine, we just get approved from Thai FDA, and we will start clinical trial phase 1 tomorrow. I'm very excited with that, and I hope if I have a chance to show you the results next time, I will have that data to show. Okay, and at Bayer Phytofarm, we hope that we can develop the technology that can produce the vaccine for Thai people and the people in our region. And this is going to be the health security, vaccine security against emerging disease for our region. Okay, at Bayer Phytofarm, we grow the plant of life. Thank you very much, and I'm happy to answer all of your questions.
Okay, thank you, Dr. Waranyo, for your presentation. Can you hear me?
Yes,
okay.
Yes.
Thank you for your presentation. It's interesting and new knowledge for me because we can use plant to synthesize or produce vaccine or another, for example, something like a vaccine. As you know, now is COVID-19. We need vaccine as fast as possible. And you saw us just in 10 minutes, 10 days, we can produce vaccine until the last step. Very interesting for me. And then actually there is some questions come to Q&A section.
Okay.
Now we come to Q&A section. It's okay?
Okay,
okay. Yes. Okay, for maybe
do you want me to go
I
this?
have to read some question from our audience because they type them in Q&A question. For the first one from Natasha Pujikosasi, my question is what how effective the plant vaccine which produced from plant using plant to kill COVID-19, and what are the advantages and disadvantages using this method compared to other methods? Please.
Yes, okay. Actually, right now, actually you can use several plants, like to produce proteins, okay? But in the drug industry, in the vaccine industry, right now, most companies use this kind of plants. It's like if you want to produce protein from bacteria, you use E. coli. It's kind of like standard platform to use, I think. But you can try using several plants. It's okay. But for this plant, the advantage of using it is quite quick to grow. Like only 1 to 2 months, you can use the plant to produce protein, right? If you choose a plant that takes like 6 months, it takes longer time for you, right, to produce new protein. And this plant is very easy to infiltrate. It's easy for the agrobacterium to transfer the gene into the plant. Then this is like the advantages. Yes. Is that?
Thank you.
Am I answered?
Please. Okay. I think for the first question is okay, and then the next question is. Sorry, you should I read it for you or you answer it by Q&A?
Yes,
it's okay
actually,
if I read it
can.
for you? Sorry?
I can read it. I saw it. Yeah.
Okay.
Actually, another question is I think it's the same same as the first one, right? Can you change to use other plant in the same family?
Yes.
Like if you search the paper, there's the paper using several types of plants. Tomato, potato. Lettuce, like you can use other types of the plant. Yeah, the question. How long it takes to plant until it is ready to harvest and use? Actually, it's around 1 to 2 months before you can use the plant. But actually, it depends on what proteins you produce. Some protein you need the young plant, some protein you need the old plant. You need to optimize that. Okay. Another question, what is the challenge to make plant vaccine? This is good question. What is the benefit between plant vaccine and synthetic vaccines? Okay. I think with the vaccines, there's different types, like different DNA vaccine, mRNA vaccine, protein, or even inactivated vaccine, but for me, I think like no vaccine that work for all diseases, like that is different. Then I think we just have to produce and try, and see whether we can get the effective one or not. Okay. But for the challenge, I would say like it might be the new platform in Thailand or even in several countries, then the challenge is how you communicate with the FDA, the regulator, to understand your technology and allow you to test it in human. That's the thing. But right now there are several countries, there are several companies using this platform to produce the vaccine already. Then and even in Thailand, we will start the phase 1 tomorrow. Then I think this is another big step for us. Okay, another questions. What do you think? Do they have different activity of the vaccine product from plant and from other platform? Actually right now for the subunit vaccine, if you heard the company Novavax, that is the protein vaccine or the subunit vaccine for COVID-19, but they use insect cells as the host to produce, for me, I think all of the subunit vaccine, the activity going to be the same, but the different activity comes from the adjuvant. That different company use different adjuvant. Adjuvant is a compound that can use to induce immune response. And this is what we have to try. You have to do experiment and see the result. Yes. Okay. Yes, that's my answer. Okay. Another one. I would like to ask you about your vaccine trial on the model organism. My question is, did you use any intervention to make positive viruses infection? Okay, that's good question. Then for all of the immune response that I show you, the antibody titer, that for all of that study, I did not challenge with the virus. Then we did not use the live virus. Okay, we just test with how when we inject the vaccine into the animals like mice and monkeys, how high of the immune response the animal can produce, right? But to continue for clinical trial, we have to prove that our vaccine can protect the animal from the challenge, right? Then we have, but like for the mice, the normal one, we cannot use it to challenge because the mice doesn't have the ACE2 receptor for SARS-CoV-2. Then if we challenge the mice with the virus, the mice cannot develop the disease. We have to use the transgenic mice that have human ACE2 in the mice, and we did try that in the mice, and we can show that our vaccine can protect the mice from the disease. Yes. Then, but for the monkeys, we did not do viral challenge because we don't have the, when we work with the SARS-CoV-2 virus, we have to have the BSL-3 laboratory, biosafety level 3. We have that for the mouse, but we don't have the facility for monkey yet in Thailand. Then we cannot try it. Okay, I think I answered that. Another questions. Okay, I would like to ask 2 questions. First one, when we use plant to produce protein, do we have to use specific plant or we can use all plant? I would say like you can use several types of plant, I cannot say all plant or not. You have to try it, How to make sure that the plant that we have choose, okay, the plant that we have choose to produce protein could produce protein that we want. Then first of all, you need to try small scale, like you have to get the plant, grow it and try it with 1 or more plant, and see whether this plant can produce the proteins or not. The second one, what about the possibility of COVID-19 vaccine, which is produced from plant when applied? Like if you ask me, I would say that there is the possibility, right? I am the one who developed that. Then I have to have hope that it will work, right? But we will go to phase 1, which means we have to test for the safety first. And if we found that the vaccine safe in human, then we can continue phase 2 to test whether it can induce immune response in human before going to phase 3. You know, like all of this is science, and we have to do the study to answer that. You know, like although we get the good result in animal, but it doesn't mean that we're gonna get the same result in human, right? Human and animals is different. Then that's why we have to do the study. It's quite interesting because we use tobacco plants. Yes. What the advantage compare with Arabidopsis? Okay. Many people use Arabidopsis, you know, but with Arabidopsis, I think most people use the seed, right, as the protein production. You know, like produce protein in seed, you get very small seed and very small amount of the protein. But using tobacco plant, you get big leaf, you know, like then in case that you want to get more proteins, I think like using tobacco plant, you can have more of biomaterial to extract the vaccine. Okay, another question. I have the question about vaccine. What do you think? Do you have different activities of the vaccine product from plant and other platform? Okay, then compare the plant vaccine with other platform. You mean like if we have to continue the study and finish phase 3 clinical trial, in that way we can compare, because the efficacy of each vaccine that you know right now, that's all the efficacy after phase 3 clinical trial. You know, then right now we cannot compare our vaccine with other platforms. The next project, is it possible to make edible vaccine in the future? Actually, you know what? This technology starts with the idea of edible vaccines, but producing edible vaccine, it leads to the concerns, like in many countries, transgenic plant is still big concern, you know. And if you produce like the vaccine in banana, let's say, you know, people worry whether they go to supermarket and whether these are the vaccine or the normal banana, right? Then to kind of like stop this kind of concern, and that's why we use the plant that is not it's not the feed. You know, we cannot eat, right? No one eat tobacco, right? Then no one concerned about eating this, right? Yeah. But you know, like if you can make it, that would be great, right? In your study, do you identify the side effect of the plant vaccine in animal study? Yes, we have to measure all of the side effect, you know, the safety in monkey. We have to see whether after we inject the vaccine, we check the central nervous system, the blood, or the behavior of the monkey, even the redness of the site of injection, whether that happened. We have to test all of that, yeah. And we did not see the side effect in animals. That's why we can continue testing in human. I'm not sure I have enough time to answer. If I run out of time, please stop me, okay? How do you put a bacteria into the plant? We use agrobacterium infiltration, you know, like use the vacuum to push the agrobacterium into the plant, okay? How do you know how much dose you have to add into the plant before extracting the protein? We have to optimize to see the dose, okay? Will it disturb the growing process of the plant? We did not see that, but you know, it might affect. But we grow it. We need the plant to grow for only 4 to 7 days. Okay, then that's not affect that much. Okay. Okay, the question I want to ask you about the issue of the transgenic plant method can damage environment. How about your opinion? Yeah, is it safe for environment? Then with our technology, we did not grow the plant outside. You know, we have to have the control environment. We grow in the control room, okay? And we did not leave all of these plant and agrobacterium outside. And I think this, with our technology, we did not affect anything for the environment, okay? How about preliminary test for the potential of the plant vaccine? Yes, we get the good data, we can show that our plant vaccine can induce immune response in mice and monkey and can protect the mice from the viral challenge. Then yeah, that's what we have right now. I think if some of you, like if the student here or even the faculty members are, interested in this technology, like, you can ask Dr. Atsa, right? She is my previous PhD student, and right now she's work in the university in Indonesia, and I think she knows the technology very well, and, I am happy if you have any questions for me, you can email me, okay? And or for some of you or some of the students, if you are interested in like joining my lab for master or PhD, I'm happy for that as well.
Thank you so much, Kachan. And then, is it okay if I have a question for you? Actually, this is, as I said before, this is new for me. Is it okay if I ask about, you said maybe we can say we cannot use all of plant as the medium for produce like vaccine, but you said before just several types. Let me know about the types of specific types of plant we can use in your research.
Actually, like if there are several plants that you can try, okay? It depends. If you want to use transient expression, you need to find some plant that you can infiltrate the agrobacterium into it, right? And those plants has to be like easy to accept the gene, but if you want to do transgenic plant, that's another story, right? There are several researchers produce a protein in tomato, potato, like banana, several plants, like you can try it, and I'm sure that for plant biotechnology we can have like use almost all of the plant, but the question is, can you scale up? Can you use it in the real world? In the industry scale. I think that's the important thing.
Okay. Thank you very much. And then I think we have one participant, Miss Chiquita. Do you want to ask directly to Dr. Waranyo? Miss Chiquita? We can open? No? Oh. I'm sorry. This is Miss. Maybe she cannot ask directly. One more question about vaccine you produce in Thailand. Have you tried in human in Thailand only, or have you spread them to other countries?
For the phase 1 clinical trial, we do only in Thailand, because for phase 1 we use very small amount of people. We use only 16 people per arms, After we pass phase 1, we will do phase 2 to test the efficacy in Thailand. But, like if we pass that, for clinical trial phase 3, to see the efficacy of the vaccine for protect the disease, we will find the site in other countries as well, and maybe in Indonesia. If we see that the vaccine is safe and have the efficacy, like from phase 1 and phase 2 in Thailand, to continue phase 3, we have to go to other countries, because we need more volunteers, like more than 10,000, people, yeah.
Okay, thank you. Thank you very much for your presentation, and I think hopefully in the next time we can have a more potential collaboration, not only in section like now, share with us about your research, but also we can we can come to your laboratory to do research, or you can come to our laboratory to we can join to maybe do research and produce some paper like that.
I think after if the COVID situation is better,
yeah.
I am
Yeah.
I'm very happy to welcome you all to my lab, you know, and I'm happy and I hope we can have the future collaboration.
Thank you very much. Thank you very much, Professor Waranyo. And so we can after this we can, because you already gave us presentation and answer all of questions. So thank you very much and
thank you.
Enjoy your weekend. See you next time. So if you want to stay here, this is okay. But if you have something like that, you can leave it. So okay. Now we can continue to the next speaker. We have here, Associate Professor Samima. Are you ready with us? Yeah, okay. Before Dr. Samima share with us about his research, let me introduce Professor Dr. Samima like before. The second speaker in this section is Associate Professor Dr. Samima binti Abdul Rahman. She is one of lecturers in Department of Pharmaceutical Science, Faculty of Pharmacy, University of Cyberjaya. She is graduated from University of Putra, Malaysia. And today, she will deliver her presentation entitled Drug Discovery in Development, a preliminary study of erythroxylum cuneatum leaf alkaloid extract. Professor Samima, are you ready with your presentation?
Yes, okay. Can you all hear me?
Okay, I can hear you clearly. Okay, right. Dr. Samima, the time is yours.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and good morning everyone.
Good morning.
So first of all, thank you to the moderator for a brief introduction of myself. And I also would like to express my thank you to the organizing committee and conference committee for inviting me to give this speech or lecture this morning. All right. So this is actually a sharing on my and our research on the plant Erythroxylum cuneatum. So this is the presentation outline where we'll be having an overview of drug discovery and development phases and the justification, of course, on how and why we need alternative for new drugs or new alternative. And of course, the plant interest is Erythroxylum cuneatum. So I'll be also talking about this Erythroxylum cuneatum, what is it? And gone through this presentation, we'll be having a discussion on the objective, the methods, what I'm doing in this research, the results and conclusion. Okay, so I know that the previous speakers have actually gone through these slides before on drug discovery and development phases, but we have actually preclinical phases and also clinical phases in the drug development, and it might be taking quite some times after the drug is being marketed in this in the world or in our country. But the things in drug development process is the crucial part is the phases where the drug is screened and identification and investigation of the novel compound or the novel properties of this particular compound or drugs or medicinal plant. Okay, so we have in vitro and in vivo testing. Okay, a preliminary study in the preclinical phases before we go for the clinical phases. Okay, and when we talk about drug discovery, the process, it will actually taking quite some times and many steps and stages. And towards the end of the day, from the very basic research where many of the compounds have been screened, then maybe those that is success in the preclinical phases maybe less than 5 or maybe 1. Okay, and at the end of the day, the approved drug is only 1 compound after a very long journey. Because of what? Because of we want to basically we want to ensure that those compounds that we screen is having that particular properties, okay, in treating diseases, and of course it is not causing harmful effects to human, to us. Okay, so that's why the drug discovery process is actually a very tedious phases. It takes many steps and stages, whereby at the end of that, the successful story is actually the one that is approved to become a drug. Okay, and be marketed and used in our community. All right, so I am actually working in this preliminary study of erythroxylum. Is our intention is to look into the anti-inflammatory effects of that erythroxylum cuneatum. So when we talk about anti-inflammatory, just take a look of what is inflammation. So inflammation is a supportive mechanism against tissue damage or infections, where we know that inflammation will actually stimulate our immune system, right, to repair the tissue, okay, to defend our body against diseases, against virus, against bacteria, against any pathogens, okay. And sometimes inflammatory reaction will lead to self-injury, okay, by provoking defensive action against the body cells, all right, or it may be exerting reaction to the less harmful antigen. So in inflammation, injured or swelled cells will produce signaling mechanism such as cytokines, all right. Then it will triggering lymphocytes. So molecularly, when we talk about inflammation, it is actually related to our immune system and how our body is react with that particular pathogen. So we know that inflammation is having two types or two kinds of inflammation. It can be either acute or chronic, but in this preliminary study, my intention is actually to look on into acute inflammation that have been induced, okay, in our preclinical study, all right. So justification of new drug for inflammation, okay, xanthine oxidase is actually the enzymes that trigger or become the indicator for the pathological disease which lead to certain diseases such as gout, inflammation, heart failure, stroke, atherosclerosis, even diabetes, hypertension, and of course, rheumatoid arthritis. And most of the existing drugs currently is actually working on to inhibit this enzyme. Okay, so inhibition of xanthine oxidase is accepted, and it have been proved to become the most effective treatment strategy for gout, hyperuricemia, and also renal dysfunction. And we know that currently allopurinol is the leading drug or the most effective xanthine oxidase inhibitor that is in our market and that have been used in the clinical conditions of these diseases. And of course, when we talk about our conventional drugs, they come with their pharmacological properties. At the same time, it will actually possess many the screening process. Even when we go for Google Scholar, okay, when we search for anti-inflammatory plants, okay, anti-inflammation or anti-inflammatory properties of many plants, many have actually studies have showed that bioactive natural products is actually having a potential to treat many diseases. Okay, even they also been claimed to possess xanthine oxidase inhibitory potential. All right, and some of the isolated compound or isolated constituents from natural sources are found to be potent inhibitor of xanthine oxidase, and the activity is comparable to the reference drugs, the existing drugs that we use nowadays, which is allopurinol. So this study is actually have been driven to see whether the alternative that we want to see, we want to study erythroxylum cuneatum have a potential xanthine oxidase inhibitory effects. All right, so all the medicinal chemistry plant, of course, when we talk about the chemical constituents, they rich with the secondary metabolites, the active metabolites that have a diverse pharmacological properties. Okay, the active metabolites, the secondary metabolites such as flavonoids, alkaloids, terpenes is actually those who are having or giving us the properties that we want. Okay, so in Malaysia, in folk medicine, xanthine oxidase have been used as anti-inflammatory and antipyretic agents, but there's so far a limited evidence on to prove that this plant is having anti-inflammatory properties. Thus, this study have been conducted actually to see that properties. All right. So the model that I use in my study is carrageenan-induced pore edematous. It is a general and common model to see anti-inflammatory effects of certain drugs or certain compounds. Okay, where we induce inflammation by carrageenan injection, and then we see whether there is an cascade of acute inflammation happen, and whether the plants or the compounds that we injected to that particular animal will actually reduce the inflammatory or inflammation process or response. Okay, the development of edema is actually through the injection of carrageenan, and then it will cause either acute and local inflammatory response, where at the early phase Histamine, serotonin, and bradykinin is actually the first mediators involved, and the secondary phase is actually whereby the involvement of many prostaglandins and cytokines such as IL-1, IL-10, interleukin-6, and also TNF-alpha. Okay. And this is, you can see that the plant from the slide, it is coming from Erythroxylaceae group, where in Malaysia, we know that this plant, the common name is Cinta Mula or inai-inai, and it is distributed throughout Peninsular Malaysia, Borneo with Sabah only, and this plant is also exists, and we can find it in Philippines, Myanmar, Thailand, Singapore as well, and it have been used in folk medicine and in traditionally as anti-inflammatory and antipyretic agent and tonic agent after miscarriage in Terengganu, Malaysia. So the claim of this plant is they have high antioxidant and it possesses anti-inflammatory effects, and some studies have actually in other group from UPM, they have found that this plant compound is effective for the treatment of opiate dependence, right? So as what the earlier session, the first speaker have actually mentioned on the choices of extracts and the extraction process. So we have various choices. Okay, when we go for the preclinical screening and preliminary study, either you choose a crude extract or you choose an alkaloid or certain component, or you go for a pure compound. Okay, okay. So when you screen for the crude extract and they give a positive effects, positive properties, so then it will goes to the next steps where isolation of pure compounds or alkaloids can be the choices of the screening of any pharmacological properties. So in this study, we choose alkaloids extraction for the animal study. All right, so the phases for these preliminary studies is to see for the first one, the antioxidant and also xanthine oxidase activities in vitro. Okay, so in this study, we are using DPPH, FRAP, and xanthine oxidase inhibition assay. Okay, and to investigate whether this alkaloid extract is having anti-inflammatory effects. So I use carrageenan-induced spore edema test, and we see in the histological study whether it is in line with the animal study. Okay, so this is the method for the extraction. Of course, there should be a confirmation of the alkaloid extract from Erythroxylum cuneatum. Okay, and then for the in vitro antioxidant assay, we choose DPPH and FRAP assay, a very general antioxidant assay to see at the first step of screening. And then moving on to xanthine oxidase inhibition assay, where allopurinol is used as a standard in this assay, and in vivo carrageenan-induced spore edema, where this methods is conducted in animal to see the inflammation or inflammatory inhibition, and also to see whether in H&E screening the histo part of the pore. All right, so this is the usual extraction method. We use either cold soap, or there is other option, but in erythroxylum cuneatum, we use softlet extraction to extract the crude extract of erythroxylum cuneatum before it is concentrated using evaporatory process. And then for the alkaloids extraction, it been mixed with acetic acid before fractionated into many fraction with hexane and chloroform, and that concentrated extract is been then undergone evaporation process to get the final yield of alkaloid extract of erythroxylum cuneatum. Okay, and for this process, after the extraction, the crude extract then undergone the confirmation whether we claim that it is having an alkaloid, the presence of alkaloid in the extract. So Meyer's reagent been added to the extract to see the presence of alkaloid. Then TLC, thin layer chromatography plates were applied for the next step, where then after a certain isolation, a certain process, when we go for suitable mobile phases and also solvent, then they were later sprayed with Dragon-Dove spring reagent. All right, and the presence of alkaloids in the extraction is actually based on the spot appeared in the extract, right? So then after the extraction and confirmation stage, the second stage is to see whether the extract is having antioxidant properties. So we use DPPH and FRAP, where these are actually the process where we screen using DPPH reagent together in a different concentration, and then it been read using the spectrophotometer. And for the antioxidant AC for the DPPH, ascorbic acid is actually been used as the reference standard. Okay, for the antioxidant DPPH screening, right? Then for the next one, antioxidant AC using FRAP. So we use FRAP reagent to screen whether it is having high antioxidant ferrite reducing agent properties. Okay, and as well, ascorbic acid is actually used as a comparative model for this FRAP. And from this, a standard curve have been constructed using serial dilution of ferrous sulfate heptahydrate before we compare the results whether they have ferrite reducing properties. Okay. The next phase, in vitro screening, also we want to see at first whether they have xanthine oxidase inhibition effects. So xanthine oxidase activity is actually this is the enzyme that is involved for the conversion of xanthine to uric acid. So we measure the formation of uric acid from xanthine. Okay, so the preparation of the solution and dilution being made before we add the enzyme, and then the enzymatic reaction was then being measured, okay, where we compare it with the blend, okay, and also the allopurinol as the standard. Okay, and this is the formula where how the inhibition percentage of xanthine oxidase inhibitory activity been calculated. Okay. So we have done with the first screening on the antioxidant and also xanthine oxidase properties screening. So then we move on to the animal phase, acute inflammation phase, where we use carrageenan-induced pore edema model. Okay, where from this study, have been approved by our CACU, Institutional Animal Care and Use Committee. 42 sprout dolly rats is divided into 6 groups, where the 6 group is, one group is for vehicle, second group is our gold standard or positive drugs, which is aspirin, 300 milligram per kilogram. Then various doses of erythroxylum cuneatum ranging from 5 to 50 milligram per kilogram. And before we start with the carrageenan-induced pore edema, the mice, the rats have been treated with either erythroxylum, the extract, the aspirin, or the vehicle 1 hour prior of the carrageenan injection. Okay, and carrageenan injection is by using subplantar injection, 0.2 ml of 1% weight per volume carrageenan suspension. Then the pore edema is measured every hour for 5 hours. Okay, before the rats been euthanized and and the specimen is collected. Okay, the pore is collected from the study for the next phase, for the next analysis. Okay, so let's move on to the results. So when we do extraction of the erythroxylum cuneatum, right, this is the percentage yield of alkaloid that we manage to extract is 0.12%. Okay, and this study, the yield is consistent to the previous study by the report by Al-Imam, where they showed that they extract 0.08% alkaloid from the plant. Okay, and the confirmation of the presence of alkaloid in the extract is through the Meyer's reagent. So the formation of yellow precipitate is present after Meyer's reagent was added. So it actually showing that there is an alkaloid in the extract. And next step is on the confirmation of alkaloids using DLC plates. So for this screening, the presence of alkaloids using TLC, we can see that there is 4 spots appeared on the TLC paper, and the separation is distinct and separate from each other. And when we talk about the solvent system that we use, the best solvent system used to extract alkaloid from erythroxylum cuneatum is chloroform over diethylamine 9:1. Okay. Then move on to the antioxidant assay. We can say that this is the standard where ascorbic acid results is appear in red, and for the DPPH scavenging activity for erythroxylum cuneatum is in blue. So the activity of alkaloid extracts actually was lower than the reference standard. Okay, and it's only appeared as effective as the highest concentration we use. Okay, 10,000 microgram per ml. And the higher EC50 value will actually indicate the lower antioxidant activity of the alkaloid extract. So we can see that ascorbic acid is actually having higher antioxidant properties compared to our extract, alkaloid extract, erythroxylum cuneatum. Okay, and it might be due to the lower potency of the alkaloids. All right, the FRAP for the FRAP assay. So this is the FRAP value where we can see that ascorbic acid is actually having a higher FRAP value compared to our plant there. Okay, and from the standard curve, when we extrapolate the properties for FRAP assay, we can say that the EC50 for the erythroxylum cuneatum is far away from our gold standard ascorbic acid, which is it implied that the alkaloid extract was less potent than our gold standard here, ascorbic acid. Okay, although its activity was lower than the reference standard for every compact concentration, but we can say that for the FRAP activity, the ferrate reducing ability of this alkaloids extract from erythroxylum cuneatum is in concentration dependent manner. Okay, next for the xanthine oxidase inhibitory assay, this is the graph showing the percentage of xanthine oxidase inhibitory activity of erythroxylum cuneatum. All right, we can see that allopurinol is having very potent xanthine oxidase inhibitory effects, where we can also see erythroxylum cuneatum alkaloid extract is having the inhibitory effects as well, but it is very mild, very low. Okay, where you can say that erythroxylum cuneatum leaf alkaloid extract was not able to show promising effect as the xanthine oxidase inhibitor in dehydroxylation pathway. So it is less competent compared to allopurinol. But the study is actually similar as the study what have been done by Saleh et al. In 2012, right? Then for the animal model in carrageenan-induced spore edema, despite of the different group that we have, okay, we can see that the leading is actually aspirin. Aspirin is actually showing a very significant decrease or inhibition of the edema compared to the rest. But we can say that erythroxylum cuneatum for the carrageenan-induced spore edema is also having an anti-inflammatory properties as well at 50 milligram per kilogram. Okay, and this is the gross photograph of the spore edema of various groups that is snapped at the 5th hour after the induction. Okay, we can see that this is the vehicle group, group 1, group 2 is aspirin. You can see that it's very, very effective as an anti-inflammatory agent. And we can see also there is a reduction over the, when we increase the dose of erythroxylum cuneatum alkaloid extract in this study. And group 6 is actually showing a significant reduction of inflammation in this study. Right, this is also the diagram showing the percentage of inhibition when we calculate it or when we measure it. Right, and group 6 is the best is allopurinol, okay, aspirin as the anti-inflammatory agent, 3.92% inhibition. The second one is erythroxylum cuneatum at the dose of 50 micro mg per kg with 29.33%. So it shows that the crude alkaloid of erythroxylum cunetum did possesses an anti-inflammatory properties from the animal study. So we can say that the development of pore edema is a biphasic event where we can see that there is the conversion from the first phase to the second phase. Okay, and there is from the results that we test, the reduction of pore edema is actually happening in the second phase of the inflammation, okay, inflammatory, and it might be due to the inhibition of the prostaglandin, serotonin, and histamine as well. Okay, and the results is actually demonstrated that the alkaloid extracts anti-inflammatory properties is better than the aqueous extract from the previous researcher. Okay, and this the findings in animal model for the in vivo study is in line with the histology analysis, where we can see that okay, the black arrow is actually showing the infiltration of leukocytes. So this is the vehicle group, the induction group, okay, and this is the aspirin group. There is a very a significant reduction of the leukocyte infiltration compared to the the negative control group, and same goes to group 6, where it can actually show a less massive infiltration of leukocytes, right, compared to group 1, group 3, and group 4 as well. Okay, we can see that there is a slight improvement and also less massive infiltration for group 5 as well. But the best is actually the highest concentration of erythroxylum cunetum alkali extract. Okay, and the H&E analysis show a consistent results as what is shown in the animal model, right? Where we can say that aspirin group is the best, but there is promising results or anti-inflammatory effects of group 5 and group 6 as well. So we can say that erythroxylum cunetum crude alkali is also showing an anti-inflammatory effects, and it is implied with the inhibition of leukocyte infiltration. Okay. So as the discussion to wrap our preliminary screening of erythroxylum cunetum, so the TLC plate showed 4 spots of alkaloids in the extract, and this might be due to the presence of the principal alkaloid there, where we have alpha-6 beta dibenzoyl oxytropane. Okay, and in DPPH free radical scavenging assay, we can see that there is an antioxidant properties exert in the alkaloids, even though it is not as good as the reference. Okay, but we can say that it can scavenge actually the reactive oxygen species. Okay, it also shows the similar result from FRAP, and the pre-treatment of rats with erythroxylum cunetum alkaloid leaf extract lead to the decrease of leukocyte infiltration and also collagen tissue disruption, and it is shown in a dose-dependent manner. So when we increase the dose, the effect is actually more better and promising. Okay, so it looks like we need to increase the dose for the future study. So as the conclusion, erythroxylum cunetum leaf alkaloid extract did display antioxidant properties as well as anti-inflammatory effects. And it is actually this study as a preliminary screening at least, an additional justification from the folk medicine claim, okay, and it can be proposed as a potential anti-inflammatory agent in the future. So I bet this is the publication from the preliminary screening, where we have the phytochemical screening as well for this research, and also the anti-inflammatory part. And I would like to thank our university for financially supported the research. My students here that is actually working hard to conduct this research, and our Department of Pharmaceutical Sciences, the one that is actually conducting all the natural products for drug discovery and development niche research in our faculty and our university. So with that, thank you to all for your attention.
Amima, for your presentation. And now, is it okay? Okay. Now we are in Q&A section for presentation of Dr. Samima. Here there is in Q&A part from you got one question from Dian Anugrahas Bulla. She asked about how did you extract the alkaloid from the plant, and how about the sedative and hallucinative effect? Did you test the effect of them? And thank you for your question. Please, Dr. Samima.
All right, how do I extract the crude alkali extract? Is we are using various solvent system. Okay, from the crude extract, we go for what we call as the more polar system for the extraction of alkaloids, where from the slides that I've shown before, that the first phase is to extract the crude alkaloids, crude extract. Then the second phase is for the extraction of the alkaloids from the erythroxylum cuneatum. We use fractionation with hexane and chloroform. Okay, before that, it been mixed with acetic acid overnight. So with the solvent system that is more polar, is having a good alkaloid yield from the previous study. So that's why we go for hexane and chloroform for the alkaloid extraction. Okay, that is the answer for the first one. The second one, the question is
did you evaluate another, say for example hallucinative effect? Because here you just present about anti-inflammation effect. He want to know, did you evaluate other effect like anti-hallucination or something like that?
Okay, so the study is actually being conducted by our group of researchers from UPM. Okay, we are from the same research group. Okay, where one student have actually go through for the not for the hallucination and sedative, is more of on the screening of the morphine tolerance, right? So we see there is an animal model if I can share later the site for the paper, if you want to further know what are the animal models that they use for the sedative and addiction. Okay, there is many behavioral study for the neuropharmacology part. All right. So one of it is actually behavioral tests, open field tests, and so on. Okay, and there are also many behavioral testing or model that can be used to see the neuropharmacology part, especially for the sedative and addiction part. If in the previous study, in my study, to see pain, we use hot plate test. Okay, for the neuropharmacology study for anxiety, we can see we can use force swimming test, tail suspension test. There's many animal models that we can actually use for the neuropharmacology model.
Thank you, Dr. Samima. If someone participants has another question, please you can raise your hand or type here, so we can read your question. But maybe at the moment I want to ask you about some question. One of them about your research. You show us about your research using erythroxylon cuneatum in primary study, and you show us the activity, antioxidant activity is too low when you use the PPH and PRAF method. Have you tried using another standard maybe, not using ascorbic acid? I mean,
yes.
Have you tried using pure alkaloid? I'm not sure what is that for pure alkaloid, but maybe I just want to know, have you tried to use another standard to compare similar kind of alkaloid in your research?
Okay, that's a very good question. Thank you, Dr. Nurmaya, for the question. Yes, we can actually use other gold standard or reference beside ascorbic acid for our antioxidant properties or our antioxidant assay. But the gold standard is actually ascorbic acid. Most general gold standard when we see for the antioxidant is using vitamin C or ascorbic acid. Okay, we can also use other alternative or other standards, but what we have in our lab is actually ascorbic acid, and the justification we don't have we screen for the antioxidant properties, and it shows positive results even though it is lesser compared to the gold standard, but as long as it shows an antioxidant, so then we proceed to the next steps. Okay, we don't change or to redo because the antioxidant assay being conducted in a triplicate manner. Okay, so it show a consistent results. But we suspected that we need to increase the concentration to see a better antioxidant effects.
Okay.
That is my answer. Thank you.
Okay. Can I give you one more question? Because we have around 10 minutes, 10 more minutes in Q&A section. My next question is about activity. In TLC result, you show us you use chloroform diethylamine 9:1 to show us there is alkaloid in your isolation, right? Did you use different kind of solvent for develop this? I mean, maybe you can show us more than 4, maybe to I just want to know, did you try to use another kind of developing solvent to show us about the amount of the number of alkaloid maybe in your extract? Not only chloroform diethylamine, but also maybe another developing solvent, or just one like this?
Okay, when we talk about the conformation of the alkaloid in the TLC plate, we can play for the solvent, right? And the best results that shows a distinctive alkaloid spots is by using 9-to-1 diethylamine and hexane.
I see.
I use my student did use actually the first screening is we use many solvent system, but the one that is separating the spots in TLC, the best one is by using this system. Chloroform diethylamine.
Okay, we have 5 more minutes. One more question, maybe the last question for this question, for this section, still come from me. Okay, about the maximum concentration did you use in to evaluate the anti-inflammation effect of your extract? Here you use 50 milligram per kilogram of alkaloid leaf extract, and this is the best result. Did you try using higher concentration for evaluate the anti-inflammation effect for this? And well, this is how to know this is safe or not. Maybe you have to continue this research. Maybe you have planned for in the future. This is in title. The title of your presentation is preliminary study. Maybe in the future, maybe you can share to us what is your plan to continue this research, because here you conclude that this extract has anti-inflammation effect, and you can continue it for the next. So maybe it is good if you share to us maybe what is your plan for in the future, and maybe we can do collaboration, because as you know, in our university we have pharmacology department. They studied about they studied is anti-inflammatory effect like this. So maybe we can do collaboration or something like that. So if you want, you can share to us actually what is your next future to continue your research about this extract, maybe to know about anti-inflammatory effect. Please, Dr. Sayama.
All right, thank you, Dr. Numaya. Okay, so for the future planning, since this is a preliminary screening of the alkaloids from Erythroxylum cuneatum, we are actually looking forward for the isolation of pure compound, because the screening have shown that it is having an anti-inflammatory properties. So the future planning is actually to investigate the pure compound that is giving the anti-inflammatory effects or anti-inflammatory properties from erythroxylum cuneatum. So this is actually the future planning or future collaboration is with chemists for the isolation part using NMR and mass spec and so on. So then we are going to the animal study as well, but we are still finding tune and also discussing, or we want to see whether which dose actually is the best dose to proceed, and which dose is actually having the not the effective dose, but we will screen for the toxicology analysis for this extract and also compound. Okay, the previous study from our group, study group, research group, they have actually screened for the MTT toxicology study. Okay, but for the future study, we'll be actually investigating the toxicological analysis on animal model as well, right? That is the future planning. Might be for our collaboration, we can work on this together, inshallah, with Umi. So maybe we will have a further discussion in the future meeting, inshallah. A very good suggestion and looking forward for that, Dr. Maya.
Your presentation, and after now I think this is enough for the third section. After two speakers, Dr. Waranyo and Dr. Samima from Thailand and Malaysia. Thank you very much joining us today. And then I want to say, please forgive me, apologize me if I made mistake. I know I made mistake during I moderated this section, so please apologize me. And then for all of attendants, please enjoy until the end of this conference. And then for the next, I pass the section to the host. Thank you.
Ladies and gentlemen, before we move to the next event, we can take a break. And for all participants, you can join back into the Zoom with us at 1 p.m. Central Indonesia time or +8 GMT. Thank you very much and see you at the time. Ladies and gentlemen, before we move to the next event, we can take a break. And for all participants, you can join back into the Zoom with us at 1 p.m. Central Indonesia time or +8 GMT. Thank you very much and see you at the time.
Angin memiriku pasar, pitujui tong-tonganan, tu saro ataka lupa, batu mi aing memiri, angin ngerang dingin-dingin, nama lantang sari buku. Aule, Angin melirik ke pasar, pitujui tong-tongan. Angin memiriku pasan pitujui tong-tonganan tu saro ataka lupa batumi angin memiri angin ngerang dingin-dingin nama lantang sari buku
Ladies and gentlemen, thank you for joining us again. Now, we will continue the webinar and the third session will be guided by Apotheker Abdul Malik PhD. To Mr. Apotheker Abdul Malik PhD, the time is yours.
Okay, thanks MC. Hello everyone. Assalamualaikum warahmatullahi wabarakatuh. Good day for Indonesian time and good morning for Copenhagen time. So first of all, I would like to thanks all of you who joined this the first international conference, Makassar International Conference, and of course special thanks to Kenneth. Hello, Kenneth.
Good morning.
Do you hear me?
Yes, can you hear me?
Yes. Okay. Special thanks to you because you accept our invitation. That this is our 2 decades of Faculty of Pharmacy. And Kenneth is academic staff at Natural Product Research, Department of Drug Design and Pharmacology, Faculty of Health and Medical Sciences, the University of Copenhagen, Denmark. And his research focus is about the anti-diabetic drugs, specific in type 2 diabetes, and the source of substances from endophytic fungi. This like something very interesting source of chemical compound. Then we have 30 minutes today. We are going to hear from Kenneth. I believe that he can bring us to a new world of microorganisms. The title is about hiding in plain sight, exploring endophytic fungi for tomorrow's medicines. So Kenneth, time is yours. Go ahead. Yeah.
Thank you very much, Abt. Thanks for the nice presentation, and thanks for allowing me to speak here today. I'm quite honored to be part of this first international conference you're hosting. So I will share my screen if I can get this to work. Just give me a second. You should think that during Corona you would have learned this by now, but it's work in progress. Share screen. Why doesn't that work? So you can probably see my presenter slide, is that correct?
Yes.
Yeah, that's not what you're supposed to see. I'll just give it another go. Oh, does not work.
Can try again.
Yeah, I'll try again. Screen sharing failed to start. Oh, excellent. At least we have 30 minutes. I will. So here we are. I hope it works now. Is this correct? You can see my presentation?
Yes, yes.
Good. All right. So yeah, first of all, thanks a lot once again for inviting me to speak here today. As Abt has already told you, I'm working at the Natural Product Research Group at University of Copenhagen, and I'm primarily interested in endophytic fungi and how they can be a source of novel chemistry and novel drug products. So today I will try to go through some of the analytical techniques we're using in the research group to be able to isolate and identify these natural products. And I will also try to, if time permits, go through a few projects highlighting some of the different approaches we're having on natural product research. So for those of you that do not know what endophytes is, A short introduction to that. So endophytes is, say, a relatively unexplored source of novel chemistry. An endophyte is a microorganism. It could be a fungi, it could be a fungus, it could be a bacteria living inside a plant, but not actually being a pathogen to the plant, but it's living in the tissue, sort of coexisting with the plants. Having access to nutrients, and often some benefits for the plants in terms of antibiotics, antifungal compounds, anti-insecticides. So how do we actually get hold of all these endophytes? Because we need to get them out of the plant material. So first thing we need to do, we need to collect some interesting plants. In this case, a small flower here. Can you see my pointer? Or should I make it a laser pointer? We'll do it a laser pointer. So we collect this plant material, and the next thing to do is to surface sterilize the plant material to kill all the epiphytes, that is all the microorganisms on the outside of the plant material. So we kill that only to be able to look at what's inside the plant. Then we cut the plant material into pieces. It could be roots, it could be branches, flowers, fruits, whatever part of the plant we're interested in. Putting it on some nutrients, and slowly the microorganisms, in that case we are targeting fungi. So the fungi will migrate out of the now dying plant material onto the nutrients we have in our agar plates. So now we just need to isolate these endophytes who have pure colonies that we can work with further on. So the approach I've been taking for the last couple of years has been based on traditional medicinal plants from Indonesia. So I had 2 PhD students, Abt being one of them. And both of these students collected traditional medicinal plants that have been used for treatment of type 2 diabetes, which has been one of our key research areas. So we collect these plant materials and isolated up to about 100 endophytes from a single collection campaign. So now we have all these endophytes. We have a lot of microorganisms that produces a plethora of natural products. So now we need to actually pinpoint which of these are active and what is the structure of these compounds. The structure of the compounds. So to do that, we have a quite unique chemical characterization platform in our research group, and this is based on analytical HPLC coupled to a high-resolution mass spectrometer. So we're doing chromatographic separation here. Then we have what is called a solid phase extraction unit. I will come into detail on what that is exactly. But this allows us to isolate compounds from the crude extract in analytical scale. So we have some trapping and some drying. And then we have a robot that pipettes this into NMR tubes. So we're using very small NMR tubes. And then we have a high field NMR spectrometer. So this is the analytical setup. And to give you a bit more insights into this part, the trapping and elution, I have a few slides on that. So here we have biomass, and we've been working with many different kinds of plant and microorganisms for a source of natural products. So here we have some K-bots. As an example, it could be a fungus. So we do an extraction, an ordinary extraction. It could be in ethanol, it could be in ethyl acetate, or whatever solvent of interest we have. Then we have an analytical scale separation. So this is a fairly simple complex mix, you could say, 3 different components. And we have a UV-based chromatogram. It could be UV, it could be MS-based chromatogram. If we're interested in the red and the yellow components here, we will set a threshold, say anything giving a UV absorbance above a fixed threshold should be isolated. And what happens is whenever a peak of interest comes above this threshold, it will be directed onto a solid phase extraction cartridge. And this is actually just a very small chromatographic column. So it could be a C18, it could be C2, whatever column fits the purpose of the particular compounds we're working with. And the essence of this is that we have this in the ordinary HPLC eluent. There's acetonitrile, there's water, and we're doing gradient run, so we have an increased amount of acetonitrile during the run. So we add a lot of water to lower the allotropic strength, the ability to actually be purged off the column. So we lower the amount of acetonitrile, and that actually allows us to retain our compounds of interest onto these cartridges. So now we have moved our compounds from a complex mixture here to a single compound on a small column. And the beauty of this is that we can do this several times, so we can up concentrate the amount of material. It will stay on the column because we add all of this water. Next thing is to get rid of all the protonated solvents we use in the analytical separation, because that really will make a lot of mess in the NMR spectrum. So we dry this with pressurized nitrogen. All this is done in automation. So we remove all the HPLC eluents, and then we elute it with the robot using deuterated solvents of interest. It could be deuterated methanol, it could be deuterated acetonitrile, whichever fits the purpose, into 1.7 millimeter NMR tubes. So they're very thin, very small. Allows us to have quite concentrated amount of material, even though we are running in analytical scale. And then we put it on the NMR spectrometer, and all that is left, you could say, is to identify the compounds. So this is a quite nice, quite efficient way of working with complex mixtures as we are as natural product chemists. So one of the things that allows us to do this, or at least makes it much easier, is that we have a 30 microliter cryoprobe installed in our 600 megahertz spectrometer. And this allows us to use 30 microliter solvents instead of the ordinary 500, 600 microliter that we use. And since it's cryocooled, we have a much higher sensitivity because of the noise reduction. So this setup is really optimized for complex mixture analysis. So enough about the analytical platform that we use for characterization of the compounds. We're also interested in bioactive compounds. As Abt already mentioned, we have had a lot of focus on type 2 diabetes. We also had a bit of focus on antibiotics. But what is common for both is that we characterize them using this high-resolution bioassay platform. So again, it's based on chromatographic separation into an analytical scale, and we microfractionate it into microtiter plates. Then we perform our bioassay of interest. And it could be whatever bioassay we have that is compatible with the 96 well plate format. And then we have some kind of plate reading that allows us to produce what we call a biochromatogram, where we can pinpoint the bioactive constituents. So in details, we have the same, we're now working with the Caber bots again. We have the same separation as we had before. And then we simply time slice it. We just slice it up into 96 wells. It could be 2 plates, it could be 3 plates, but whichever amounts of plates that make sense in terms of the chromatographic profile. We're aiming for something like 3 to 5 wells per minute to give us a high resolution. So everything goes into microtiter plates or into the wells in the microtiter plate. And then we either dry the plate, get rid of the eluents, and perform our bioassay, or we can perform it directly in the HPLC solvents, depending on the assay we are performing. So each of these wells are now time referenced to the HPLC chromatogram. So pinpointing the activity below here allows us to say, oops, here we have some activity, and here we also have some activity. So we are reversing the activity, so the activity goes down. I can see there's a question. That was the host. So so we can now pinpoint, say, okay, of these three compounds, the red and the yellow, that is the one with activity. So we only focus on that in the analysis. That is at least the purpose of this. So that was the profile that, or the analytical technique we're using. And I have two examples on how we use it, so you can actually see how it looks in real life, because this was nice and textbook examples, but how does it actually look when we go in nature and look for bioactive compounds? So the first example I will give you, that is a dual high resolution alpha-glucosidase and PCP1B, those two target enzymes in type 2 diabetes, of this fungus, Colicotridium theobrobicola, that we isolated, or actually apt isolated from Persea americana. And this has been traditionally used to treat diabetes. So what he did was he collected this plant material and surface sterilized it, as I described to you, put it on some nutrients. We use potato dextrose agar, a lot of nutrients. And then he isolated, among others, this very nice fungus. So we always run this in 3 points inoculation. So we have some confrontation zones between the fungi that allows us to get a bit more complexity in the metabolic profile, the secondary metabolic profile. So this is the fungus, and now all we need to do is to figure out, does this fungus actually contain anti-diabetic compounds? And this could, some of these anti-diabetic compounds it produces, could that actually be the reason why this plant material have been used in traditional medicinal or in traditional medicine? So performing a extraction with ethyl acetate, apt got this. You can see this is the chromatographic profile. This is UV absorbance, 254 nanometers. The beauty of fungi compared to plants is often that we have a much cleaner chromatographic profile. Fungi tends to be very focused on particular biosynthetic routes, so it produces a lot of compounds in the same pathway, whereas plants tend to produce compounds and then they post-modify it, put a lot of carbohydrates on and so on. So the chromatographic profile of fungi tends to be more clean, more crisp to see. So few compounds of interest here. So this is the alpha-glucosidase inhibition profile. This is a PCB1B inhibition profile, and as you can see, and this has nothing to do with capabilities of app, because we redid this many times. But the alpha-glucosidase inhibition profile is a bit messy. You can see we have activity corresponding to peaks, but it sort of tails out. So there's something going on in the chromatographic profile with the compounds tending to be a bit broad. And often we see this, that the activity profile, the biochromatogram, tends to be more sensitive. So we have a very nice peak up here corresponding to peak 2. So we have an activity, but then it sort of tails out, so it's more sensitive in compared to the UV detector. So this is because app did this 2 times, so we both have an alpha-glucosidase inhibition in a large enough scale, so we could do some pharmacological testing on them afterwards. So app isolated these 9 compounds. Some of them were known. These 2 are known compounds. This is known as well. The rest are new compounds. And as you know, natural product researchers and in the world of pharmacy, you know that knowing the absolute stereochemistry is quite important when we're doing drug discovery. So how do we actually? So we have isolated the compounds. NMR can tell us how they look, but how do we actually find the absolute stereochemistry? So in this case, we see if we got this on the next slide. So the absolute stereochemistry was determined using different approaches. One thing is that we found that all these compounds actually come from the same biosynthetic pathway. Give me a second. So they come from the same biosynthesis. So stereochemistry is expected to be retained throughout the biosynthetic route. So this is, I know there's a lot of compounds on this, and it's quite small slide or small structures, but what I will show you is it all starts from a polyketide synthase, which produces this, and then we have a non-ribosomal peptide synthase, so we put on a tyrosine on this polyketide chain, on with the tyrosine, and then we can go two different ways. We can either go this way up here to produce compound 5, 4, 9, and 8, where we have this this polyketide chain combined with the amino acid. And then we have different modifications on this. So these are the ones where we are a bit struggling with the stereochemistry of a lot of the carbons. But it also goes in another route. So what we saw was that we have a ring expansion here. So this ring expands, so we get this compound here, and this can either be cyclized, so the polyketide chain here is cyclized to produce either compound 1, compound 2 and 3, and compound 7. So determining the stereochemistry of, for example, compound 7, 3, 2, and this has already been done because these are known compounds. We were actually able to backtrace the stereochemistry of the methyl groups, for example, here, all the way back to the starting material, and then retain that and go into these compounds 4, 5, 6, and 8. So using the biosynthesis actually allowed us to determine the absolute stereochemistry, or at least we are quite certain that we have the absolute stereochemistry based on that. So next thing up is to confirm that hypothesis, and we did that by circular dichroism. So this is looking at plane polarized light, like a optical rotation, but it gives us a bit more information. I won't go into details on the technique, but what you can do is you can calculate the expected CD spectrum and compare that to the experimental spectrum. So these does not look exactly the same, and that is primarily because of the enormous amount of flexibility we have in this ring, in this chain here. And we also have a bit of an issue with this probably being in both epimers. But looking at this this side chain here, you can see that we have sort of the same trend in the calculated blue spectrum and the experimental orange spectrum. Same thing goes here. We have a bit of an issue here. We have some negative effect here. We have a lot of negative effect there. Some positive, a lot of positive. But besides that, it looks very similar. And this confirms that our expected absolute stereochemistry is correct. So that was determining the absolute stereochemistry. I'll just, I'm running out of time. I'm talking too much apparently. So app, let me know when I need to stop talk, right? But we also have the IC50 values because we need to find bioactive compounds. So we did IC50 values in both alpha-glucosidase and PCB1B, and I will primarily look at the PCB1B because this confirms what we saw on the biochromatogram, that only one peak actually showed activity, and that was peak number 3, compound 3. Interesting enough, compound 2 and compound 3 are actually atomizomers, so they are two ring structures that can be in different ways. So we have actually the same compound, but in two different conformers, only one being active. So this was quite interesting. So we looked into that by molecular docking studies. So this is PCB1 B enzyme, a model of that, and we have two different binding sites we're investigating. And this is the known inhibitor of this binding site that was it was crystallized with this ligand here. And when we tried to dock both compound 2 and compound 3, we found that it actually docked in the same conformation as compound 3. So compound 2 is converted into compound 3 when it docks into the binding pocket. So we observe this conversion. Okay, interesting, say. But this doesn't really explain why we only see activity for compound 3, as compound 2 would just be converted into compound 3 when we perform the assay. So to figure out if this conversion actually happens, we try to do some studies again on the computer, but we try to investigate what happens if we put a lot of energy into the system. Do we actually have enough energy to overcome the barrier? What do you call that? No, doesn't matter. To overcome the energy barrier converting from compound 2 to compound 3. And what we saw was that the angle between C3 and C7 is actually the same throughout the study. So even at 500 Kelvin, we do not produce enough energy to be able to convert from compound 2 to compound 3. So this actually shows up, this actually shows us that compound 3 is the active constituent, because compound 2 doesn't really dock correctly. So that was another approach into this. And now my time is up, so I will skip the last story I had because I talked too much. But I would like to thank the audience. I would like to thank the organizing committee for inviting me. It has been a pleasure, even though it's a bit early in the morning here in Copenhagen. And I would like to acknowledge, so this is a past picture of the research group. So we have Abt here, and he's been one of the driving forces in doing the endophytic work. And then we have Sirful, who has been working on the other project I didn't have time to come into. And then I would like to also acknowledge the LPDP for providing funding for actually having both Abt and Sirful in our research group for 3 very productive years. So with that, I will say thank you to all of you.
Okay. Thanks, Kenneth, for your very nice presentations.
You're welcome.
And we have 30 minutes for discussions. And we get one question from Nureski Khairunisai. should I read for you, Kenneth, about the question? We got
Yes, I just need to. Oh, I can find them here. Oh, I can see. So should I read them out loud?
Yeah.
Okay. So good afternoon.
From Rizky,
I'm
the first one.
Rizky, you would like to ask about the research that you perfectly explained. Thank you very much. How can we know the specific fungal endophyte can generate new metabolite compounds? So the question, if I understand it, is how do we know that the actual endophytes we are isolating has the potential to produce new compounds? And the short answer is we don't. That is the entire challenge of doing drug discovery from nature and particularly endophytes, because there's so many of fungi in general. There's so many microorganisms, and a lot of the compounds that we isolate is already known. So for sure that's a challenge, and it's I want to say it's a numbers game. So that's why we isolate a lot of endophytes, and we screen them. We do initial screen just at 2 different concentrations to see if we have any activity at all. And then the next step, I didn't show this, but what we often do is we do MS-based replication. So that means performing a high-resolution MS, or acquiring a high-resolution MS chromatogram. So we can use this to tentatively identify the compounds using high-res MS. We can have a molecular formula. We have a UV spectrum. We can search this in databases like Reaxys or a more focused database like Antibase. And then we can say, is this a potential known compound or is it a potential new compound? If it contains a lot of known compounds, I would probably just skip that fungus, go to the next one, because it's sort of trial and error. So when we do these presentations, we always find the nice examples. There's a lot of, and you can ask apt, there's a lot of fungi that doesn't really produce anything of interest, at least in the assay we're testing on. So I hope that was answer enough. If not, please just ask again.
The second?
Yes, is this from Andy Rismayanti?
Yes. How we can identify
What's sorry, Abt?
How we can identify the plants has an endophytic fungi?
Yes, that's a good question. Most plants do have endophytic fungi. There's actually very few plants that does not have endophytic fungi. There's a few examples, but most have. And it can either be pathogenic fungi that migrates into the plant and thinks, oh, it's actually very nice being here. I got plenty of nutrients. I got lots of sugar from the plant. Why should I actually kill it? So there's a few examples where we don't get any endophytes out, but that is really, really very few. Yes. So we do this in when we are teaching. We have an experiment where all our undergrad students, they go out and collect different leaves, and we plate them out, and we get a plethora of fungi from all of them. So it's just dig into it.
Kenneth, maybe before we move to the next question through the chat room, maybe direct question from Mr. Fawaz. Go ahead.
Oh, okay. Thank you so much for this opportunity. Good morning, Dr. Kenneth.
Good morning.
For local time in Denmark. So thank you so much for your presentation. I think this topic is my cup of tea. So I'd like to ask you about the molecular docking of your research. 33 PPT, the number of page.
Yeah.
So I want to know the 33 page of your presentation.
Yes. Should I share this? Can you see it?
Okay, I can see.
Can you see this now? Or do I need to share my screen again? Oh, you can't. I will share my
I
screen.
think no problem, sir, because I got your PPT file from the organizing
okay.
committee, so
Okay,
I can.
excellent.
Okay. Hi. Okay. So I want to know which part of this protein take an important role for this activity of your drug. It is the first one. And the second, I want to know, when we know the structure of that compound, the number 3, I think the structure is not so complicated. So maybe it's easy to synthesize. So what do you think about the synthesis of this compound for the future? Thank you.
That was good questions. So I must say in the short answer to your first question on which you're asking which residues is actually interacting with our ligand. Is that correct?
Yes.
Yeah. So I am. We're collaborating on this, so it's not really my area of expertise. I would love to be able to explain it, but I do not. I can't do that.
Okay.
Because we have another research group that are focused on this. I can find it out and let you know when I talk to Fleming, who did this.
Oh, okay.
Regarding the second question, that was the synthesis of these compounds. I'm just thinking, because I think they actually have been synthesized to some extent already in literature. So I think the main issue with synthesis of these is that there's quite a lot of stereo centers. So you need to be able to control that during the synthesis. That is the only challenge, in my opinion. But I think I would rather go for a more green chemistry approach, doing synthetic biology, because the beauty of fungi, in my opinion, there's a lot of beauties in fungi, but one of the beauties is that you can more or less take out the cassette of genes coding for the enzymes producing this compound and take it out. We might need to knock out a few genes to make sure that we go the right direction or modify it a bit, but then we can put it into another organism. We can put it into yeast, and then we can produce this in a high yield. So if we are not interested in doing further medicinal chemistry on it and optimize it, that might be a possibility to make it even more active. Then I would probably go for the biosynthetic production of it.
I see. Okay, I get your answer. Thank you so much for your explanation.
You're welcome.
Okay, thank you, Mr. Fawaz. Then we have one more question left from Chiquita. She asked about the relationship between endophytic fungi and plant hosts. Are they have relation in production of secondary metabolites? Yes.
Yes, and that is also an excellent question. And this is so the hypothesis is that there is a relationship. So to give you a bit of history lesson, you can say the research into endophytes really peaked after the isolation of Taxol, paclitaxel, the anti-cancer drug compound that come from the tax trees. So that's traditionally isolated from the bark of the they're called yew trees. I think it's called the big pine trees. And we actually found, researchers found an endophytic fungi living internally in these trees that was also capable of producing this blockbuster anti-cancer compound. So there was a discussion whether is it the plant producing it or the tree in this case, but is it the higher plant or is it the microorganisms? And if it's both, because it's really hard to investigate a plant without endophytes. You need to culture the plant under sterile conditions and you can say hope that there's no endophytes in the seeds. Because if there's endophytes in the seed, you already have an endophytic community within the plant material. So to make it short, we believe, and we're quite certain that there is some horizontal gene transfer between the plant and the endophytes. So the really short answer is yes. There is a correlation between it. We don't really know if it's the plant or the endophytes producing it, but we believe that since microorganisms is much more sustainable way of producing drug compounds, it makes sense to investigate if traditional medicinal plants actually contains microorganisms that can be a source of these compounds, instead of having to harvest or having big fields of particular plants. We can actually have them in a tank reactor and then just produce the amounts we need, and then use the fields for other better purposes. I hope that was an answer to that question.
Yes. Okay. Yes, one more question from Andi Maulana. He asked about a good method for get antioxidant specific compound from plants.
Yes.
You get the question.
Yeah, so antioxidant specific compounds. So it's the isolation of them, you could say if you want to identify the antioxidant
compound,
the antioxidant compounds from plants. If you can do the microfractionation, that is one of the simplest assays to perform, because you don't need to evaporate any of the HPLC eluent. You can perform the antioxidant assay directly in the microinsular plate, and this is a very neat way of pinpointing the antioxidant compounds. And I can share I can send that to Abt, so perhaps you can share that in the network. We have a very nice paper on actually how to do exactly the antioxidant assay. There's different ways of doing antioxidant assays, but if you can do microfractionation, and you can do this in different ways, you can do it both with fully automated, but you can also do it more manually. We've done that also. It's a very nice and easy way to find the antioxidant compounds. I hope that was answering that question. If not, please ask again.
Yeah. We have one. We have two more. One more question from Andi Rismayanti about the chance to get a new drug for COVID-19.
For COVID-19?
Okay.
Oh, that's a big question. If we can find new drugs for COVID-19 from fungi. Yeah, as a natural product researcher, I would say yes. I do believe that we can. I don't think it's going to be easy. You have to, at least if it's something that would actually make an impact, on COVID-19. I think we can find plenty of compounds that have some moderate activity, but if we really want to find something that is that has the potential to be a marketed drug, then we need to search for within a lot of fungi before we find it, and we need to find the right assay. So we probably need to set up various assays, testing different, using different ways of testing for the COVID-19 activity. But it's also a question: what do we want to target? The symptoms? Do we want to target the virus or whatever? Which approach will you take? Yeah, that was one thing I wanted to say also. Yeah, that's one thing, and that is you should also be aware that we have only scratched the surface in terms of fungi. We've only looked at around 5% of the fungi in the world. We looked at a lot of fungi already. So there's a lot of fungi unexplored. So it's just a question of actually getting them out of the soil, out of the plant, out of the water, and look at what do they produce. But for sure, yeah, I think there's a chance. It's a slim chance, but it's there.
Okay. I think still many questions to you, Kenneth, but time is running out, I think.
Time's running.
But I mean, if someone wants to ask again, maybe one more question, if any more from lecture from the Faculty of Pharmacy, maybe Nurmaya Effendi, or yeah, Nurmaya Effendi, yes.
Okay, thank
please
you.
go ahead.
Okay, thank you. Hello, Prof. Kenneth.
Hello.
Hello. Okay, thank you. Nice presentation.
Thank you.
Actually, I would like to know about the potential to evaluate the compound which you find from using this method, the possibility to evaluate the anti-cancer activity. You said we can actually you got some isolation from HPLC, and then we can evaluate in each well.
Yeah.
We can evaluate that using, if we want to know about anti-cancer activity, we can evaluate directly from that vet and using some kinds of human cancer.
Yes. So
yeah.
If I understand your question correctly, so whether we can do this anti-cancer activity assays on using this platform. Yeah, so if that is the question, then yes, and we are actually already moving into that area. So what you should think about when I present this high resolution biochromatograms and pinpointing activity in each wells, it's a qualitative analysis, so we can just see there's activity in this well. We don't know we don't know the amount in the wells, so we just know there's compound there, and we can see okay, it's active or it's not active. And of course, depending on the amount and depending on the activity, we'll have more or less activity when we test it. And then we need to purify it and test it afterwards, so we can make these IC50 curves, for example.
Okay.
So
it's possible.
It is possible, yes. It's a bit more difficult because it's cells, so you need to make a monolayer and you need to work under sterile conditions. Whereas for the enzyme assays, you don't need to work under sterile conditions. So it's a bit more tedious work, but it's for sure it's possible. We've done it with bacteria. We also tried it with cancer cells, and that also works. But the baseline is a bit more. There's a bit more variation in the baseline. Often we do it in triplicates to see if we can account for some of the variation we see, because it is live cells we're working with.
Okay, thank you.
You're
And
welcome.
one more question,
Yeah.
if it's okay.
Yeah.
About the possibility to make collaboration with Professor Kenneth about. I mean, in your slides, you saw us. You can find until the final structures which active, for example, as antifungal or something like that. It means it is possible to synthesize them, and after that collaborate with other collaborator, for example, and then evaluate in vivo. I mean, we use animal not only in vitro. It is possible like that, or you already have collaborator for
No,
example,
actually,
in your compound?
No, it's we have some collaboration in different areas, but we have always focused mostly on the initial drug discovery, so finding the compounds. So the in vivo studies is something we would like to do much more, but not something we have you can say been good at prioritizing. So for sure that's potential for collaborations on that.
Thank you very much.
You're welcome.
Okay, thank you for Nurmai Effendi for your questions. But I think that it's really the time now is really running out. But I mean, we still. I hope for Kenneth someday in the near future, you will come to visit us in person in Makassar in
It would be a
Indonesia
pleasure,
and
for sure.
collect some endophytic fungi. Yes. Before we close it, maybe Kenneth have the last comments, maybe. Yeah.
Not really, actually. Not much. I would just like to thank you all for inviting me. I think it's really a pleasure to be able to join conferences like this. So thank you for that. Thanks to the organizing committee and all your questions. It was nice to have this live discussion on the topic. So thanks for being active.
Okay. Yes. Okay. Thank you very much. Again, they're grateful for you, Kenneth, because for your time, and I say, I mean, maybe someday, yeah, someday. Then time is up, and I can say that have a good day, have a nice weekend, Kenneth, for you, and also all of the attendees today. So for that, that's all of the last part of our session. So now I give the stage to the MC. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. and Mrs. for hosting all the speakers. Now, we can continue the event with oral presentation. For our presenter, you can prepare and join the breakout room according your ID and your room. Thank you, Mr. and Mrs. Hosting for hosting all the speakers. Now, we can continue the event with oral presentation. For our presenter, you can prepare and join in the breakout room according your ID and your room.
oh ini
Ladies and gentlemen, now it's time to announce the best presenters. We have 8 best presenters. They are the best oral presentation, Room 1. Syazawani Mazelan, Bachelor Pharmacy. The best oral presentation, Room 2. Apoteker Asriani Suhaina, SSI MKes. The best oral presentation, Room 3. Apoteker Ahmad Najib, SSI M-Farm. The best oral presentation, Room 4. Apoteker Nur Rahma Rumata, S-Farm MSI. The best oral presentation, Room 5. Nurul Anizah, Bachelor Pharmacy. The best oral presentation, Room 6. Aina Syafia binti Hashim. The best oral presentation, Room 7. Apoteker Ana Husnul Faiza, S-Farm M-Farm Clean. And the last, the best oral presentation, Room 8. Steffi Lim. Ladies and gentlemen, thank you for joining us until the conference ended. Thank you very much to all the participants and see you next events. Wassalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, thank you for joining us until the conference ended. Thank you very much to all the participants and see you next event. Wassalamualaikum warahmatullahi wabarakatuh.